Ladies and gentlemen, welcome to the second International Conference on Genetic Resources and Biotechnology. Please introduce, I am MC for today. My name is Wening Enggariti. Our Honorable Director General of Indonesian Agency for Agricultural Research and Development, our respected directors of research with center within the IAARD and Indonesia Institute of Science, respected chairs of Indonesian Biotechnology Consortium, respected directors of partnering companies, our distinguished invited speakers, distinguished deans of universities, distinguished head of research institute within the IAARD, and all participants. Assalamualaikum warahmatullahi wabarakatuh. Good morning, good evening for everyone. We warmly welcome you to the second International Conference on Genetic Resources and Biotechnology. We are also very grateful that this year we are able to meet you all in good health in this event, which raised the topic harnessing technology for conservation and sustainable use of genetic resources for food and agriculture. This international conference is made possible through the collaboration between the Indonesian Agency of Agricultural Research and Development and the Indonesian Biotechnology Consortium, and is held virtually since we are still in the middle of the COVID-19 pandemic. Now, to start this event, let us first listen to the Indonesian national anthem, Indonesia Raya. Ladies and gentlemen, thank you. Next, the chair of the organizing committee, Dr. Toto Hadiarto, will present a report on the organization of this international conference. To Dr. Hadiarto, welcome.
Assalamualaikum warahmatullahi wabarakatuh. Honorable Director General of Indonesian Agency for Agricultural Research and Development, respected Director of Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development, respected Chair of Indonesian Biotechnology Consortium, respected Directors of Research Centers within the Indonesian Agency for Agricultural Research and Development and Indonesian Institute of Science, our distinguished invited speakers, distinguished deans of universities, distinguished directors of partnering companies, distinguished heads of research institutions within the Indonesian Agency for Agricultural Research and Development, and our enthusiastic participants. Thank you to each and every one of you for being here with us in this virtual conference. My name is Toto Hadiarto, and I'm the chair of the organizing committee for this event. On behalf of our organizing committee, let me begin by giving you a warm welcome to the second International Conference on Genetic Resources and Biotechnology. This conference is organized by Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development under the Indonesian Agency for Agricultural Research and Development in collaboration with Indonesian Biotechnology Consortium. The theme for our conference is Harnessing Technology for Conservation and Sustainable Use of Genetic Resources for Food and Agriculture. In this two-day conference, we will learn about 5 topics of effective management of conservation and sustainable use of genetic resources for food and agriculture, application of genomics and molecular markers for genetic resource conservation, and crop adaptation to climate change, application of innovative crop improvement techniques for conservation and sustainable use of plant genetic resources for food and agriculture, plant cell and tissue culture for conservation and effective utilization of genetic resources, the use of microbial genetic resources as biological control agents of agricultural pests and diseases, and for soil bioremediation. We will hear from our 5 invited speakers from different countries of Indonesia, the United States of America, Japan, and India about their expertise and knowledge on the subjects in the plenary session. This conference is attended by a total of 119 participants, including 75 presenters and 44 listeners worldwide. They come from diverse governmental, private, or academic institutions, and also from scientific communities. The presenters will share their scientific information and technological developments in 5 classes of parallel sessions. All participants will be encouraged to exchange their knowledge, ideas, and thoughts in the discussion of each session. In this conference, the presented materials have undergone peer review processes and only qualified papers were selected. Furthermore, all papers are subjected to double-blind peer review and are expected to meet scientific criteria of significance and academic excellence to be published in AIP conference proceeding series. Now, before I finish, I would like to express my gratitude to our speakers, presenters, and all participants for their contributions in this conference. I would also like to express my appreciation for the generosity of our sponsors that support this conference. They are PT Crop Life, PT ITS Science Indonesia, PT Fajar Mas Murni, and PT Prima Instrument Analytica. My special thanks to all committee members for their exceptional works to make this event happen. I hope that this event will be successful and collaboration both nationally and globally can be possibly developed. Before we begin our plenary session, I would like to invite our Honorable Director General of Indonesian Agency for Agricultural Research and Development to deliver the opening remarks and to open this conference officially. Thank you very much.
Thank you, Dr. Hadiarto. Ladies and gentlemen, the Director General of the Indonesian Agency for Agricultural Research and Development will officially open this event. We welcome Dr. Fajri Jufri to give his opening speech and open this conference.
Respected Director of the Research Center within the IRD and Indonesian Institute of Science, respected Chair of Indonesian Biotechnology Consortium, respected Director of Partnering Companies, our distinguished impact speaker, distinguished Dean of Universities, distinguished Head of Research Institute within the IRD, and all participants. Assalamualaikum warahmatullahi wabarakatuh. Good morning and good evening for all. Distinguished participants, ladies and gentlemen, it's my great pleasure to address at this international conference on genetic resource and biotechnology. This is our second conference to establish continuation of the first one in 2018. During this time, we are still facing the COVID-19 pandemic, so we are doing this conference virtually. Ladies and gentlemen, genetic resource for plant, animal, and microbe, and mankind heritage and need to be used optimally or sustainably to meet the increased demand for food and energy. Advances in biotechnology have created more opportunities for effective conservation and sustainable utilization of genetic resources for food and agriculture. This conference will discuss the current development related to these issues. However, we will limit our discussion to topic outlined in the scope on the conference. Ladies and gentlemen, make conservation and genetic resource become sustainable for the future. The future climate is also significantly changing. Therefore, this type conservation must utilize advanced technology such as genomic and molecular marker application. In order to do so, innovation of technique for crop improvement are always encouraged through research and development. Assessing and identifying new source genetic variation an important gene and essential to enhance the productivity, sustainability, and resilience of newly developed crop varieties and agricultural system. Finally, it's my sincere hope that this conference will share enhance current scientific information and technological development and biotechnology and their application for conservation and sustainable use genetic resource. And let but not technology. Bismillahirrahmanirrahim. Thank you very much.
Thank you, Dr. Fajri Jufri. We will work hard to make this event a success. And now, ladies and gentlemen, we enter the photo session. Please all participants to turn on video and give the best smile. Okay, ready? Now I count. 1, 2, and 3. Okay, thank you. Please the other page. For the second page, be ready. 1, 2, and 3. Okay, for the third, I count 1, 2, 3. Okay, the others, the fourth, I count 1, 2, 3. Okay, well done. Thanks to all. Ladies and gentlemen, now this is the end of the opening ceremony of the second International Conference on Genetic Resources and Biotechnology. We are very grateful for your presence today. Thank you very much. Ladies and gentlemen, you are welcome to take a short break before we enter the first plenary session. We will begin the session in 10 minutes from now until 8:45. Oh no, 8:15. And it will be moderated by Dr. Muhammad Yunus. So I would like to hand over the floor to Dr. Yunus, and I will see you again at the end of the first plenary session. Have fun.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya adopsi teknologi pertanian berbasis produk rekayasa genetik, dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
The Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development is a technical unit of the Indonesian Agency of Agricultural Research and Development under the Ministry of Agriculture. It aims to become a leading research institution in the field of biotechnology and genetic resources to support the development of advanced, self-reliant, and modern agriculture. ICA Biograd has 4 research groups, which are Genetic Resources Management Group, Molecular Biology, Cell and Tissue Biology and Biochemistry Group. The main tasks of Genetic Resources Management Group are to characterize, conserve, identify, and to map the physical mapping and genomic characteristics of agricultural genetic resources. The Molecular Biology Group is tasked with research in the field of agricultural biotechnology, development and application of molecular markers, and genetic engineering. The Cell and Tissue Biology Group is tasked to perform research in the field of cell and tissue culture for crop improvement and propagation, production of secondary metabolite, and in vitro plant conservation. While the Biochemistry Research Group has the task to perform research on metabolomics and proteomics from bioprospecting to downstream application using biotechnology approaches. One of the facilities to support research activity in ICGRB is the Gene Bank. The Gene Bank is a storage facility to conserve agricultural genetic resources under the international standard. It has the storage room for short-term storage, medium-term, and long-term storage, which is capable of maintaining samples for 100 years. Currently, there are more than 11,000 genetic resources stored in the Gene Bank, comprising food crops, horticultural crops, estate crops, insect and microbes. Other facilities that are available in ICGRB are laboratories, limited testing facility, foundation seed production unit, greenhouses, experimental facility, and many more. To respond to the fourth industrial revolution, ICGRB has developed a central information system in the form of the Indonesian Agricultural Genome Center and the Gene Bank database. The Indonesian Agriculture Genome Center is the first genomic database in Indonesia. It has been granted intellectual property rights, while the Gene Bank database stores information on conserved genetic resources and services to deposit and access genetic resources. To fulfill strategic national program and anticipate consumer demand, ICGRB actively produce new variety of food crops, horticultural crops, estate crop, and livestock feed crops. Some of the released food crop variety are Impari Blas, Impari HDB, Impari 40, Biopatenggang, Biodi 63 Ciherang, Padi Gogo Bio Bestari, and Padi Bio Salin. Farmers in many regions have utilized them because they are excellent yield, resistant to blast, resistant to bacterial light blight, and resistant to brown plant hoppers. ICGRB also released BioSoy soybean variety. It has a large seed size and weight 20 grams per 100 seed with the potential to produce 3.5 tons of seed per hectare. BioSoy is currently being planted in several provinces to boost national soybean production. ICGRB has also released Bioguma sweet sorghum variety, which can potentially produce 9.3 tons yield per hectare. It is being utilized now by several regional government, farmer, and private sector. In the effort to improve the yield and quality of horticultural product, ICGRB is developing the Garvey Agri Horti Red Chili Pepper. It is resistant to stripe virus and can produce fresh chili from 22 to 30 tons per hectare. Aside from new plant variety, ICGRB also developed various technology such as tissue culture technique, pheromones, early maturing sugar palm detection kit, non-certified oil palm seedling detection system, and pregnancy test kit for livestock. Tissue culture technology has been developed for various crops such as for garlic, amorphophallus, bananas, orchid, sugarcane, and potatoes. ICGRB also carries the mandate to provide recommendation during the drafting of regulation related to biotechnology and agricultural genetic resources, hosting the National Commission for Genetic Resources, technical team for biosafety of livestock feed, and to act as the focal point for various international organizations such as ICGRB, HLPDAB, and Genetically Modified Food Network.
Ladies and gentlemen, may I have your attention, please? First of all, we would like to apologize for the technical problem before, and the presentation is about to begin. Honorable invited speakers, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Good morning, Ohayo gozaimasu, and Magandang Umaga. First of all, I would like to welcome you all to today's plenary session of the ICGRB 2. My name is Muhammad Yunus, and I will be moderating this session, and I am glad that you are all here. In this plenary session, we will have 3 invited speakers that are Professor Michael J. Thompson from Texas A&M University, Professor Hiroshi Ezura from University of Tsukuba, and Dr. Mastur from ICGRB. In this session, we will first listen to the presentation, and the remaining time will be used for discussion session. We will have enough time for this plenary session that is about 2 hours and 20 minutes. But before we start, I would like to ask Professor Thompson, because we have a different significant time. It is night time in Texas. Which one you prefer to have discussion after your presentation, or later after we finish all of the presentation and then we have a panel discussion after that? Yeah, we consider, because we are worried it will be very late in Texas.
Oh, it's okay. I can join. Okay. I can stay the whole session for the panel talk. Is okay.
Okay. So we will have all of the presentation first, and then we will continue with the panel discussion. And for the participant, if you have any question to the presenter, you can type the question in the chat box or ask it directly when the discussion session is open. Okay, let's start the presentation. I would like to introduce briefly our first speaker, that is Professor Michael J. Thompson. He graduated from Cornell University in 2002 for his PhD degree in plant breeding, and now he is a professor of the Department of Soil and Crop Science in Texas A&M University and the head of the Texas A&M AgriLife Research Crop Genome Editing. And the title of his presentation is CRISPR-Cas9 Genome Editing for Crop Improvement. Professor Thompson, please, time is yours.
Okay, let me share my screen. So can you see the presentation?
Yes.
Okay.
Yes, Mike.
Great. Well, thank you very much for the invitation, and it's really my pleasure to give this talk. And I am talking live from Texas, so if you have any issues with audio or video, just send a message in the chat and we'll see if we can fix it. But today I will be discussing CRISPR-Cas9 genome editing for crop improvement. And as was mentioned, I'm currently at Texas A&M. I've been here about 5 years. And before that, I was at Erie in the Philippines doing rice research. And we're still using rice as a model system for our genome editing. And it's been a number of years since I've been in Bogor, but hopefully next time we can do this in person, but it's still a wonderful opportunity to have a virtual conference as well. So I will go ahead and get started. So today I'll be discussing really an introduction to what is CRISPR-based gene editing, and moving on to how it can be used for crop improvement, and then how we're setting up really a high throughput gene editing pipeline at Texas A&M in the crop genome editing lab. And then I'll finish with some challenges for commercialization of how the regulatory process is moving, especially here in the US. So just for those who are not as familiar, CRISPR is really a breakthrough gene editing technology. And I put the acronym here, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. So it's, of course, CRISPR for short. And really in 2012 was when this technology kind of came to the forefront. And this was a publication from Jennifer Doudna and Dr. Charpentier, who are working together on this technology. And actually last year they won the Nobel Prize for chemistry due to the CRISPR-Cas9 breakthrough. And so today we'll focus on that. And so just a number of advances with CRISPR-Cas9. First, that it essentially forms molecular scissors that can cut DNA at a specific location. So what we're looking at here is the Cas9 protein. And so you'll see it can make a double strand break. So it can cut both strands of the DNA at a specific location. And this is guided by an RNA molecule. So this is a single guide RNA that really kind of forms a complex with the Cas9 protein. And this really is a much easier system than previous gene editing technologies. And so it's actually very easy to set up in the lab and to get up and running. And so that's really why it's become so popular in many fields of science. And if you look at, it's in the news, one of the more controversial areas, of course, is editing. So human germline editing. But of course, in agriculture, it's a lot less controversial. And you'll see that there's a large number of publications now using CRISPR technology, and also a large number of patents. And so the intellectual property also is kind of skyrocketing because it is a very valuable technology. And so it's right now impacting a wide range of fields, from basic biology to medical research, human therapeutics, virus, bacteria, plant, and animal research. So it's really become kind of a hot topic recently. Okay, so just looking a little bit about the history. So I mentioned 2012 was kind of the landmark paper, but CRISPR biology had been around for a lot longer than that. Really back into even the 80s, 1987, the first CRISPR sequence motif was described. And then, in the past 20 years, there's many publications trying to describe what essentially, where did CRISPR come from, and what is the mechanism, what is the function. And in parallel to that, there was also the field of genome editing. And early on, there's many different genome editing technologies. The most famous are the zinc finger proteins. And then after that, the TAL effectors and talons being used for genome editing. And it wasn't really until 2012 that CRISPR biology and genome editing came together in that, again, that landmark paper. And since then, it's really exploded in the field. So from 2013 onwards, we have papers in many different species using this. It's accumulating these segments from previous viral infections, and that's then passed on to the progeny. And so the bacterial populations will essentially have a memory of previous viral infections. And what that does is it allows this immune system to react really faster when the next virus comes, it will instantly recognize that viral genetic material, and then it will cut the invading viruses. So that's how the bacteria essentially can defend themselves against these bacteriophages that are the viruses attacking those bacteria. And the key part here is essentially there's the initial recognition here is called adaptation, where the bacteria captures that genetic material and then it produces a CRISPR RNA. And then targeting is where it recognizes the new viral infection. So this is important because the Cas9 protein is what's involved in both the targeting and the double strand break with that viral genetic material. So just looking a little bit closer, what we have is the CRISPR system has a number of different proteins. Some of them are involved in adaptation and creating this CRISPR repeat spacer array. And some, like Cas9, is involved with the recognition and cleavage of the DNA. And so you'll have, again, this CRISPR repeat spacer has the viral sequences, and then those will be used to form a complex with Cas9, and that is what then will recognize and make the double strand break. And so what we're seeing here is that essentially a 20 base pair sequence is the recognition sequence. And you have essentially at the bottom, you can see that 20 base pairs. I didn't mention it, but there's also a PAM sequence involved. So a 3 base pair motif that has to essentially be directly adjacent to that 20 base pair recognition sequence. And then the double strand break is 3 bases upstream of the PAM motif. And so that's where the DNA will be cleaved by the Cas9 protein. So here's a picture of the Cas9 protein surrounding the DNA. And essentially, that's where it makes that double strand break. Okay, so when people talk about using CRISPR-Cas9 genome editing in plants, essentially the Cas9 will make the double strand break, but the plant cell will actually repair that DNA. And essentially there's 2 different repair mechanisms in plants. One is non-homologous end joining, or NHEJ, and the other is homology-directed recombination, or HR, or HDR. And essentially these are the 2 repair mechanisms. And so when CRISPR-Cas9 cleaves the DNA and makes a double strand break, the most common repair mechanism is the non-homologous end joining. And so the plant will actually essentially ligate those ends together and it'll fix the double strand break. But what happens is that's a very error-prone process. And so it won't connect them perfectly. Often there's 1 or 2 base pairs either deleted or inserted. And if that occurs in the coding sequence, that will actually knock out that gene function. That's usually due to a frame shift mutation that causes a premature stop codon, which can cause a truncated protein. And so even just a single a double strand break at a single site in the DNA can cause a gene knockout. Another really mechanism is you can add a template DNA. So if you add donor DNA as a template, sometimes it can be inserted at the locus if you have 2 double strand breaks. Sorry. And the other is homology-directed recombination. And so that's where you have, again, there's a DNA template, but in this case, it uses recombination to correct or fix the double strand break. And so you could insert, a single base change, or you can even insert a large segment, and those will either be a gene modification or a gene insertion. And so these are essentially all the different mechanisms of repair and also some of the different applications of genome editing. Okay, so in plants, of course, many of you are familiar with genetic engineering. And so this is also used for genome editing as well. So for instance, you have to, in most cases, create a transgenic plant. But first, what we need to do is design the guide RNA. And so you need a target gene. And then once you have the gene sequence, you can choose a 20 base pair target sequence adjacent to the PAM, which for Cas9 is NGG. So it's any base plus GG. And that will serve as your guide RNA sequence. And so you have, essentially, you can insert that into this guide RNA backbone. And you need an RNA promoter, such as U6 or U3, to express the RNA in your plant sequence or plant genome. and that will create the guide RNA. And at the same time, we also need the Cas9 to be expressed. And so what most people do will combine the Cas9 and the guide RNA on a single transformation construct or plasmid, and then deliver those into the plants. And then the Cas9 will interact with the guide RNA to make the double-strand break. And so, of course, those of you involved in genetic engineering, you can use you can transform protoplasts. You can do agrobacterium-mediated transformation. You can also do callus bombardment using the gene gun, so biolistic transformation. And there's a few other ways as well. But the key point is you need to express both the Cas9 protein and the guide RNA at the same time inside the cell for it to be active and to make the desired mutation. And the last step really is to screen your transgenic plants for the gene editing events. And there's many different ways to do that. One is you can design the cut site at a restriction enzyme site, and then it will essentially disrupt the recognition motif and will cause a loss in that restriction enzyme activity. There's also the cell 1 or T7 assay, or you could just sequence the target directly. Currently, agrobacterium is still probably the most common technique for this. Okay, and I did want to mention, especially here in the US, that AdGene is a very popular service, and it's essentially a nonprofit plasmid repository. And so if you're interested to start gene editing in plants, you can actually just request the plasmids from this service. And of course, with the MTA involved, you can get them delivered. So you can get the Cas9, you can get different promoters, and also different, essentially, now there's a lot of different variations in nucleases. So in addition to Cas9, there's CPF1, which is also called Cas12a. So there's a lot of different nucleases now available. And this service actually makes it much easier to get started. So once you have the plasmids, the next step is really to design the guide RNA. And there's a lot of online tools to help with this. I'm just showing one called CRISPR Direct. And the key point here is that essentially you need the 20 base pair recognition sequence next to your PAM motif. But at the same time, you don't want, of course, to be multi-copy. You want a single copy recognition site in the genome, because we want to avoid off-target effects where you cut essentially a locus that is undesirable. You just essentially want to cut only your target locus. And so these online tools actually can compare your sequence across the entire genome. And really, it just checks for potential off-target effects. So for instance, for rice, it will compare against the Nibombare sequence. There's now some indica sequences available as well, so that you can choose kind of the essentially the most efficient guide RNA design that will have the fewest off-target cuts. Okay, and so essentially, I did want to talk about a few of the newer technologies. So what we talked about just now, say agrobacterium, you have delivery and integration into the genome. But there's another method called ribonucleoprotein delivery, which is RNP delivery. And essentially that is just where you deliver the purified Cas9 protein in a complex with the single guide RNA, and there's no DNA involved. So essentially it's called DNA-free genome editing. And you can use, say, biolistics to shoot that complex into the plant cells. And if they do get into the nucleus, then they can be expressed and make the desired mutation. And so this is often used as a way to get non-GMO or non-genetically engineered plants by using RNP-based delivery. And related to that is you can actually take the Cas9 itself and attach fusion proteins to it. And what people do now is you can deactivate the cut sites. And so there's two essentially nuclease cut sites, or essentially the place where the Cas9 will cut the DNA, and those can then be deactivated. So it'll be a deactivated Cas9, so it won't make a double strand break anymore. And then you can attach a transcriptional repressor or transcriptional activator, or even GFP or demethylase. And now there's even nucleases that target RNA as well as DNA. And so each of these provides another method mostly for functional genomics to essentially assay like gene function by overexpressing or underexpressing the gene expression. And I will mention another recent technology that's quite exciting. It's called prime editing. And prime editing, it also uses Cas9, but it uses a Cas9 nickase, which means it will only make a single strand cut. And then it has a prime editing guide RNA that then will include essentially a sequence that will be used as part of the repair mechanism attached to the guide RNA. And so what that allows actually is for all the different point mutations and small insertion deletions to be precisely edited in your target. And so that kind of avoids the process of Cas9, which normally is a very messy process, so it's not very precise. So prime editing essentially would be much more precise. And so that's something that many groups are now testing. There's some publications in plants, especially in rice, where they show that this is successful in rice and other plants. Okay, so that was just the introduction. I did want to go into some of the strategies for crop improvement using genome editing. And I just show this as an example of some of the related technologies. So I think most of us are familiar with the genetic diversity that is available. So crop genetic resources, and also the gene discovery allele mining activities. And so we have essentially this useful pool of alleles and genes. And there's what I see as kind of three different strategies for plant breeding to use this, essentially to use biotechnology. The first is marker-assisted selection, and now genomic selection as well. So high throughput SMP genotyping to select for large effect genes or for across the whole genome for complex traits. And of course, that will be a non-genetically modified product. So it's a non-GMO. On the other side of the spectrum, of course, is genetic engineering, where you use biotechnology to introduce transgenes. And so that would be transgenic product. And so that is GMO. But right in the middle is genome editing. And so we can use CRISPR technology to make these precise changes. And depending on the method used, it can be actually non-GMO or it can be GMO. Depending on, for instance, if you integrate Cas9, you can actually get rid of it by segregating it away in the final generation. And so you can have a non-GMO product at the end of the day. So that in some ways makes it more powerful, I think, than many of the previous methods. Okay, so I'll just run through a few publications using rice as an example. And so this is one of the early ones actually just did a pilot study to knock out the phytoene desaturase gene, or PDS, and that gives this albino kind of dwarf phenotype. And you'll see here that, again, they just express Cas9 with a single guide RNA. And then they use, in this case, this restriction digest site to track which one of these actually was mutated. And again, you have usually one or two base pair insertions or deletions that will knock out that gene function. And if you think about how can we use this for plant breeding, so knockouts themselves sometimes provide a desired phenotype. And so two examples in rice, one is SD1, semi-dwarf one. If you knock out the GA20 oxidase gene, it will actually cause semi-dwarf plant height. And another one is the BAD-H2. If you knock out this gene, it actually causes aroma or fragrance in rice. And so those are two genes where if you knock out the gene function, you can actually get the desired phenotype. But in many cases, knocking out the gene will cause undesirable phenotypes. And so then you may need to do more precise modifications, so amino acid substitutions, or even allele replacement, where you maybe delete the old gene and then insert a new allele. And in some ways, this could be used to replace marker-assisted backcrossing because we can now do multiplex gene editing to essentially insert these novel alleles into the genome. And just one example, so multiplex editing is when you target more than one locus at the same time. And so there's actually a publication from 2017 where they targeted 8 loci in rice and were able to come up with different knockouts, different combinations of these 8 genes that were knocked out. And some of these affect plant architecture and others affect grain size. And so you get this kind of range of diversity from knocking out some of these major genes in rice. And that just shows some of the power of the technology because you can knock them all out essentially at the same time. Now, I mentioned that knockouts are not always useful. And so sometimes it's better to do precise insertions, replacements, or substitutions. And again, this requires, again, a donor DNA template. So when you target, you can target an insertion, you can delete the target and do a replacement, or you can just add single base substitutions to have very precise modifications. And these are ways then that we can essentially modify the alleles at different genetic loci. And just thinking about, what can we do in rice? Because rice has such a large amount of genetic diversity. So many of you may be familiar at IRI, they have the 3000 Rice Genomes project, where they sequence 3000 rice genomes. And they have that allele information online from IRI. So there's a Rice SNP-seq database. And if you go in, you can actually look at many of these major genes that have been well characterized in rice. And so you may be familiar with many of these. So for instance, bacteria blight resistance, like XA5, even salinity, SKC1, and then this is grain shape or heading date. Many of these large effect major genes are well known. And if you go into the database, you can get a number of different functional alleles at each of these loci. And we call those functional nucleotide polymorphisms. And previously we were able to select for those using markers, but now we can actually use gene editing to actually replace the alleles in your target germplasm. And so as long as you know the gene sequence and you know essentially the desired allele, potentially you can use that for gene editing as a fast track system instead of marker-assisted backcrossing at that point. So now I want to move into some of the recent work that we've done at Texas A&M and the Crop Genome Editing Lab. And we have a project that we call CRISPR Editing for Healthy Foods and Resilient Crops, where there was an internal project program at Texas A&M called the X-Grant project or program. And we were awarded one of the X-Grant awards to test this system. And so the first step really is to develop what we call kind of an informatics pipeline for selecting your gene targets. And then we have a high throughput CRISPR delivery, regeneration and validation pipeline, and then using trait engineering to improve specific traits. And so the long-term goals, of course, is better human health, increased global food security, and then having stress resilient crops for sustainable agriculture. And I'll just run through some of our recent activities. We actually work very closely with the multi-crop transformation facility at Texas A&M. And so this facility does the tissue culture and regeneration for each crop. And so they spend a lot of time for instance, cotton, potatoes, sorghum. Recently we did rice using a Texas rice variety. And essentially we work with them for all of our agrobacterium mediated transformation. And we're also essentially testing some other delivery approaches. One is using protoplast isolation, really as a kind of a fast track method to test your guide RNA function. Because with protoplasts, it's very simple to transform them with Cas9 guide RNA and see, essentially sequence, extract DNA and then sequence the protoplast to see if you have your successful mutations. And then another more recent technique is actually trying to transform the shoot apical meristem tissue. So we tried this in rice, where we expose the shoot apical meristem and then use that with the gene gun to bombard with plasmids to try essentially, again, a fast track method to avoid essentially the lengthy tissue culture process. So if there's a way to essentially transform the meristem tissue directly, then let the seedlings grow, it'd be essentially a faster process. So that's still ongoing at the moment. We did try a few new technologies on campus in the engineering department. They actually have room temperature plasma jets, which essentially is a very high voltage run through, in this case, helium gas. So the helium gas, along with the extremely high voltage, creates this room temperature plasma And we're essentially trying to use that as a way to deliver the CRISPR reagents. And the thinking here is that it's similar to electroporation in that the electricity, the electric field will actually cause pores to open in the cells, and then we can transfer the CRISPR-Cas9 plasmids into the plant cells. And we're using fluorescent reporter genes to see if that was successful. But again, that's still also ongoing project. Another more recent one is using nanoparticles for delivery, and in this case, carbon nanotubes. And this is some research out of UC Berkeley, Marquita Landry's lab, where in 2019, they published several papers discussing the use of single-walled carbon nanotubes. And what they did is they were able to attach plasmid DNA to the carbon nanotubes using PEI as essentially as an electrostatic attraction to attach the plasmid DNA to the outside of the nanotubes. And what happened is those nanotubes then can essentially passively diffuse past the cell wall and into the nucleus of the plant cells. And so we essentially took that protocol, and now we're testing it in a number of different crops. So rice, sorghum, peanut, and cowpea, to see if we can use carbon nanotubes for what's called implanta transformation. And again, trying to bypass the lengthy tissue culture phase so that you don't have to go through, say, 9 months of tissue culture regeneration. You can actually do it kind of in the fast track method again. And so just kind of a few examples on the traits. So my interest in the past has been salinity tolerance. And so I had a graduate student working on essentially characterizing the HKT family of sodium potassium transporters. And for those familiar with salinity tolerance, there's actually 7 members of the HKT family in rice. And this family is actually involved with sodium transport into and out of the xylem, so loading and unloading. And this actually is involved with transport of sodium throughout the plant as well. And so this is important for many different salinity tolerance mechanisms in rice. And those of you who have heard about this, so there's like 7 or 8 different members. And the problem is it's very difficult to functionally characterize a multi-gene family because you may have a knockout of maybe 1 member, but then there's still 6 or 7 members that are functional. And so it's hard to essentially identify the function of each of those gene members. And so the idea here is that we can use gene editing to make knockout combinations of essentially all the genes, or for instance, all the genes minus 1, or different combinations of these gene members. And so that will help us characterize the function, because then we'll know, essentially if we knock out all, but just leave 1 functional, then we'll know the function of that remaining gene. Okay, so again, that's an ongoing project. I'll mention another one that actually Dr. Septiningsi, many of you know Septi, she's working on actually resistant starch in rice, and so knocking out the starch branching enzymes. And there's actually 4 of them in rice, and so this is an important pathway for amylose. So the proportion of amylose versus amylopectin in the endosperm. And so this is something that previously people have started to knock out these genes, but we haven't yet seen what happens when you knock out, say, 3 of them or 4 of them. How does that affect the amylose content in rice? And I will mention one example, one way to use the RNP that I mentioned earlier is there's an in vitro RNP assay that can confirm the activity of the guide RNAs. Essentially, you just use a amplicon from PCR and then use an RNP with your guide RNA in vitro and see if it can cut your PCR amplicon. So it can all be done in a test tube in vitro. And then you have the cut versus uncut, and that will determine if your guide RNA is active to cut that target DNA. And so that's just one example from our lab. So just kind of moving on, really the final part of my talk is looking at challenges for future commercialization of gene-edited crops. And if you think about really the regulatory aspect, In the US, there's actually a recent rule that we call the Secure Rule. This is coming out of APHIS, which is from the United States Department of Agriculture. So that's the agency involved with approving transgenic crops. And so the Secure Rule is called Sustainable, Ecological, Consistent, Uniform, Responsible, Efficient Rule. And essentially what this does is it defines a set of exemptions for gene-edited plants that were modified in a manner that could otherwise be achieved through conventional breeding. And what that means is if you have a mutation that could have happened naturally, then they're not going to regulate it as a transgenic crop. So as long as it doesn't have any foreign DNA that's integrated, and as long as your mutation falls under one of these three categories, it'll be exempt from any of the transgenic regulations, and so it does not have to be deregulated. And so what that means is if you have, for instance, a change if it's already in the same species and you just take the entire gene and move it to another plant in the same gene pool, then that's essentially exempt as well, as long as it's a known allele of that gene. So you can't make any changes in the gene, but if you just take a known allele and transfer it from one variety to another, then it will be exempt under these rules. So what that means is that, at least in the US now, we can do gene editing, and as long as we essentially get rid of any of the foreign DNA insertions, then we can plant them in the field, and we can use them of course, for research and development, and then they can be more easily commercialized because they do not have to be deregulated, essentially. And so that, of course, if they're a food crop, you still need FDA approval. So that's the Food and Drug Administration. And so there are other agencies involved. But at least for planting, the crops in the field, it is much easier now because of these new rules. But I will mention that it does not include multiplex editing. And so these deletions, essentially it's a single target deletion. And again, it's also a single base pair substitution. So you can't do multiple substitutions as well. So it's really just the very basic mutations that are covered under these exemptions at the moment. So we can do multiplex editing in the lab for research, but we can't yet, for instance, grow them in the field or commercialize them unless they fall under one of these exemptions. Okay. Now, Europe is a very different story because many of you may know a few years back that there was this ruling in 2018 from the European Court of Justice that essentially said all CRISPR gene editing will be considered as a GMO. No matter, if the final product is transgenic or not, as long as it's gone through that CRISPR system, it will have to go through the same extensive regulatory process as GMOs. And of course, European scientists, not very happy about that because it's very strict regulation of CRISPR crops. But I've heard some people talking about how this may change in the future. But for now, as far as I know, it's essentially still somewhat of a moratorium on gene editing because it's lumped together with GMOs in Europe. Okay, so in addition to the regulatory system, there's also, you have to consider the IP landscape. So the intellectual property, and a few years back, there was this announcement, DuPont Pioneer and the Broad Institute joined forces to essentially enable licensing of CRISPR in agriculture. And now DuPont Pioneer is Corteva. But this essentially provides an easier pathway if you're going to commercialize. Essentially, anyone who will sell a gene-edited crop needs to license the technology. And in this case, there's a kind of a one-stop shop for licensing technology. And those of you may also be familiar, the Broad Institute at MIT was in a patent dispute with UC Berkeley, where they both had different patents on CRISPR technologies. And as far as I know, that both patents have been, are still standing, that they both have their own patents. And so potentially people may need to cross-license with both of them, or depending on the situation, maybe choose one or the other. So that's really most of what I had to talk about today. Just to conclude, CRISPR-Cas9 really has tremendous potential to make a large impact in crop improvement. Really, we can use the knowledge in genomics to modify these genetic pathways and introduce novel traits. The technology continues to improve, and we'll see more advances in the future. And the IP situation and regulations is becoming more clear, at least in the US. On the regulatory side, it's becoming more clear recently. But I will mention that consumer acceptance will be essential, because if people think of gene editing as the same as GMOs, there's quite a bit of anti-GMO movements around the world, and so that could be an issue as far as consumer acceptance. But I'll just show a picture of our lab. So Septi and I are kind of co-leading this lab, and this was a picture from about 1.5 years ago before the pandemic. So of course we're not wearing masks, and many of these people have already moved on, so we don't have a recent picture. But we are quite happy at Texas A&M and with the crop genome editing lab that we have going forward. Okay, so thank you very much.
Okay, thank you, Professor Thompson, for the informative and interesting presentation, as always. And then we now continue with the second presentation that will be delivered by Professor Hiroshi Ezura. I will introduce him briefly. Professor Ezura graduated from Hokkaido University in 1993 for his PhD degree in horticultural science. At present, he is a professor of Faculty of Life and Environmental Sciences, University of Tsukuba, and the director of Tsukuba Plant Innovation Research Center, University Tsukuba. And the title of his presentation is Gene-Edited Tomato for Human Health. Professor Ezura, please, time is yours.
Okay, can you hear me? Can you hear me?
Sorry?
Can you hear me? Hello?
Yeah.
Can you hear me?
Yeah, yeah.
Okay, okay.
Yeah, we
okay,
can
I
hear you.
would like
Okay.
to start my presentation today.
Okay.
And first of
Okay,
all,
please.
and thank you very much for inviting me to this very, very important conference in your countries. And I'm very happy to present our current, maybe it's a very, very hot topics for the application of CRISPR technology for the improvement of a crop. And can I share my slide? Okay. Okay, so you can see my slide. Can I start my presentation? Okay, so I would like to start my presentation now. Okay. Can you hear me? I'm sharing my slide so far, and can you hear me?
Yeah, it's okay.
It's
Professor
okay.
Ezura, yeah, we can hear
Okay.
very well.
Okay. Okay. Thank you very much for inviting me to this important conference in Indonesia. And I have been to visit Indonesia many times, and I have many friends in Indonesia. And so I'm very happy to present our recent research on the application of CRISPR gene editing technology for the improvement of crop. And I think that this is today's topic is I would like to introduce the application of this technique for the improvement of tomato, and which is I think becomes the first gene-edited crop commercialized all over the world, which is produced by CRISPR-Cas9 techniques. That actually this month we started providing the seedling to the consumers this month. So today's my topic is gene edited tomato for human health, and my name is Hiroshi Ezura from University of Tsukuba, Japan, and I'm currently director of Tsukuba Plant Innovation Research Center, and I was also professor of Faculty of Life and Environmental Sciences of the universities.
Ini didahulukan.
Okay, so this is the content of today's content of my presentation, and which include the four content. First one, I would like to introduce some Japanese project for crop improvement, which carried out for last 5 years in Japan, which is application of gene editing technology for the improvement of agriculture crops. We call it SIP. And the second topic is the background of gene editing research in tomato. And today, and the third topic is the gene edited tomato for nutritional quality improvement, which is we increase the amino acid content in tomato by CRISPR-Cas9 techniques. And finally, I would like to introduce the current situation toward the social implementation of high GABA tomato, CRISPR tomato in Japan. Okay, and the first, I move on to the first topic now. So this slide shows the national project, which is supported by Japanese government, which is named the Cross-Ministerial Strategic Innovation Promotion Program. We just abbreviate it is SIP project, which is supported by the Cabinet Office of Japanese Prime Minister, and which is funded for 2014 to 2018. And in this project, we try to apply the gene editing techniques for the improvement of agriculture crops and some fishes. And this project I did for this project, and then I divided this consortium into 4 groups. One is a group one is to developing a new or improve the gene editing technology, and also the group 2 is finding out the target genes for gene editing. And the third group is which I also read, it's apply gene editing breeding techniques for the improvement of different kind of agriculture products, which include tomato and rice and potato and some fishes. And then group 4 is social implementation consortium. And in this project, we simultaneously did how to the study how to improve the social implementation or public acceptance to these new techniques. That is a key point in this project. And then we spent 5 years. And then we developed a variety of potential crops to be commercialized, which is a super high yield rice. We could increase 20% of yield by this gene editing techniques. And also potato with a low natural toxin. We knock out the natural toxin genes and then reduce the natural toxin like solanine and the chaconine in potato. And also we engineer the blue fin tuna fish for easy aquaculture system. And then my group did the high quality and performance tomato using this technique. And today I'm going to introduce these topics because this is already start commercialization in Japan. Okay, so the second topic, background of gene edited research in tomato. In the next slide, I would like to introduce somehow the background of this development. And my group is studying the important breeding trait, how to regulate it, molecular mechanism of important breeding trait in tomato using a mutation breeding systems. And then important breeding trait is include like a shelf life and also fruit setting. That is a parthenocarpic fruit setting, which we can set the fruits even in hot temperature condition in tomato fruit. And also sugar accumulation and the nutrition like GABA, which is today's topic, and the lycopene and vitamin, how to introduce those nutrition in tomato plants. And these are topics we are studying so far. And yeah, here is this slide briefly show what we are doing for the mutation breeding. We created mutants lines by the chemical mutagenesis mainly using this small tomato we call a microtome. And then we try to find a mutation with mutants with a mutation in the breeding trait. Then once we found the mutants, then we isolate the genes responsible for the mutation. And currently we are keeping more than 20,000 mutant lines in tomato, which is the largest mutant correction all over the world based on a single tomato cultivars. But anyway, through this mutant breeding, we found several interesting mutants, and also we identify the genes for the mutation. So here you can see the example. This is the parthenocarpic mutants showing which we can this mutant can set the fruits even under the hot temperature condition. Tomato cannot set the fruits more than 35 degrees centigrade, but this mutant line can set the fruits even such a harsh condition. And then we identify the genes of with a mutation. Then finally, we found the mutation in the DERA genes, which is responsible for gibberellin sensitivity in plants. And then we know that a single mutation in this gene can induce a parthenocarpic, provide a parthenocarpic phenotype, as you can see here. And also we found another mutants line showing a long shelf life. These mutants can keep the fruits for almost 2 months. It's like this. We keep the fruits after harvesting for 60 days at the room temperature. This is a wild type, and this is a mutant line. The mutant line show still looks fine. And then we try to identify the mutation, and finally we got the mutation in the ethylene mutation in the ethylene receptor genes. Just a single base changes T2A. And these single base changes extensively improve the shelf life of the tomato. So through this, these two are examples, but through this research, we found that the small mutation can improve the performance of tomato plants. So then in this mutation breeding, we use a random mutagenesis, but as I mentioned in the previous speaker, we had a new technology like a gene editing in the 2012 CRISPR-Cas9. Then we try to use this, create the same mutation using this CRISPR-Cas9 techniques. Okay, so this is a background why we are interested in using this new technique for the improvement of this crop. And then in the third topics, I would like to introduce an example of the application of CRISPR-Cas9 techniques to the improvement of nutritional quality of tomato. Okay, so this is maybe everybody know that the say that the world population is going to increase, as you can see here. Then up to 2050, the population will be almost 10 billion people. So that's why we need to increase the food production and the animal production in the next 30 years. So this is our global issue in the field of food sciences. But here you can see the population changes in Japan. Unfortunately, Japanese population was a peak in the 2010, and then after 2010, we are losing population as you can see here. And then during this situation, we are maybe discussing about which kind of food is useful for this in our society. So here you can see the change of population structure in Japan, and then 1975 to 2000 and 2025, and we expect 2050, and we will have a lot of aging people. And during this situation, we have a lot of people suffering from the lifestyle-related diseases. So then one solution is to improve our health through a daily meals or daily food. So that's our mission is to provide a nutritional, high-quality food for this for overcoming such kind of lifestyle-related diseases in Japan so far. So, and then among the trait we are studying, and then we try to increase the GABA. This is an amino acid, which is well known for a kind of a functional amino acid for promoting human health. Here you can see the amino acid, but GABA is the full name of GABA is gamma aminobutyric acid, GABA. At the GABA in human, here you can see the function as a neurotransmitter and reduce blood pressures and also make induce a relaxation because of this characteristic of function. So many Japanese food companies use this GABA for their products, and here you can see some example of health promoting functional food, which is already commercialized in Japan, such as GABA enriched chocolate. It is very, very familiar in so far Japanese market. And then we focused on this amino acid. And then my group is spend a lot of time to understanding how tomato accumulate the GABA in the tomato fruits. And then here you can see the GABA metabolic pathway in higher plants. This is a common metabolic pathway in GABA accumulation, GABA metabolism in plants. And then we try to find which is understanding which is the key genes regulating the GABA accumulation. And then finally, we identified several genes for GABA synthesis and degradation. And then eventually we found that this SL-GAD3 is the main target for gene tag CRISPR-Cas9 for increasing the GABA accumulation. And I didn't. I will skip the detailed mechanism. So this enzyme, which has a auto-inhibitory domain at the C-terminal end, and then if we delete the C-terminal end, the protein produced from this deleted genes show a higher enzyme activity that's resulting in higher GABA accumulation. So then we saw that when we can introduce a mutation at this site by CRISPR-Cas9, like a stop codon mutation, then we can create a high GABA. We expected we can create a high GABA tomato fruits. Then we did using a CRISPR-Cas9. I don't need to explain this more detail, already explained by previous presenter. And then we use this technique, and then we introduced the mutation at the terminal at this site by the CRISPR-Cas9, and then introduce a stop codon mutation. As you can see here, those mutants cannot produce a AID domain in the enzyme. So then here you can see the result of the GABA accumulation in the fruits. Wild type is the original one line, and these are the 53 and the I1, or those line is a created line with CRISPR-Cas9 without the auto-inhibitory domain. And as you can see here, GABA accumulation increased to 7 to 15 times more than the wild type, just introducing one single mutation into these genes. Okay, and after that, we did this experiment in the model tomato species, and then after that, we tested whether the high GABA line can use for a tomato breeding line, because the tomato are mostly used as a F1 hybrid. So that's why we are also expecting the phenotype created by CRISPR-Cas9 must be a dominant trait. So that's why we made a high GABA tomato line, and we make a F1 hybrid with other pure line parental line. They made a F1 hybrid line. Then we measure, characterize the GABA accumulation in this F1 hybrid line in tomato. And as we expected, those line, control line, and F1 hybrid line made by high GABA tomato parental line, it shows same ripening performance, and also only GABA accumulation is increased in the F1 hybrid. So that means that indicates that this trait is a dominant trait, and also there is a minimum effect on the other metabolic amino acid metabolism in this plant. So, and here you can see the change of GABA accumulation during the fruit development, mature green stages, breaker stages, and red ripe stages where we can harvest fruits. And this white bar show a control, and then these two line, it is a gene edited line. It show 4 to 5 times higher GABA accumulation, which is almost 200 milligram per 100 gram fresh weight. Okay, and so these results show that the high GABA tomato line can be used for a parental line for F1 hybrid production in tomato breeding program. Then the conclusion is the tomato which has a GAD3 without the autoinhibitory domain by gene editing showed highly increased GABA level, which leads to breeding goal to 800 to 160 milligram per fresh weight. And the line generated by gene editing techniques were useful as a potential line for hybrid breeding. And in conclusion, this gene editing technology is useful for rapid and precise breeding in high GABA hybrid tomato breeding. Then this is a POC, a proof of concept experiment using a model tomato cultivars. And then I'm sorry, some slide in Japanese, but as I mentioned in previous speaker, to commercially use this CRISPR-Cas9 technique, we need to get a license for the licensor. And then so our university cannot generally get the license from the, because university is on, the main mission is education and research, so basic research. So that's why to commercialize our techniques, I developed the university-oriented venture company. The name is the Sanatex companies, and then this company get license from the for the CRISPR-Cas9 techniques, and then we start the commercialization of these new products. And then here you can see that commercial cultivar we established by using CRISPR-Cas9 technology, and then now has been we developed, and we started the commercialization on just 12 days ago, May 12 this year in Japan. The cultivar name is Sicilian Rouge High GABA. This is the first CRISPR crop commercialized all over the world, I think. And then so we are now we start providing the seedlings to the home gardeners. We thought that if we use a routine system to produce a tomato, like a farmer produce a tomato, and then vendor buys the tomato, and then finally consumer get. But this route is difficult to use in the original cultivar, original CRISPR-Cas9. We wanted to directly provide this technology to the consumer. So, and we decided to provide these lines to home gardeners. And then, surprisingly, we finished the notification that our products to the Japanese government last December 11. And then we announced that we are going to provide these new tomato cultivar with high GABA as a free monitor samples to home gardeners. And then initially we thought maybe we have an application, just 500, 600 application, but surprisingly we had more than 5,000 application. Then this month we start delivering these seedlings. Here you can see some packages. We provide the 4 seedlings and some other fertilizers, and we deliver to this gene-edited tomato to our home gardeners. And then now they already started growing in their home garden, and then they can harvest the fruits maybe early July, and then they can feel and they can taste those our high GABA tomato. Okay, so and then finally, I would like to somehow introduce the towards social implementation of high GABA tomato in Japan, including the regulation of this gene-edited crop. Okay, so to commercialization, we need to make several issues to be clear. And we started our original project, as I mentioned, in the 2014. In the 2014, Japanese government do not have any regulation rule about the products of gene-edited crop, but which is a national project. And then government simultaneously started the discussion of how to handle how to use these products created by gene editing. And then they can finalize the Japanese rules in the 2019. And also the to commercialize, we need to discuss about how to handling of intellectual property rights, and also improvement of social license or social acceptance. And for the Japan, this slide, you know that the gene editing technology is divided into 3 classes. One is SDN1, just cutting by, and then repairing is by natural, followed by natural repair, and the SDN2, and we need a template, and the SDN3, we can introduce additional genes. And in general, the Japanese rule is if we produce crops by SDN1 technology, that will be excluded from GMO and considered to be a normal plants. But if we produce SDN2 and SDN3 gene editing technology, that must be considered as a GMO. So this is a brief definition rules in Japanese government. But for commercialization, we need to notify the two ministry in Japan. One is the Ministry of Agriculture and Food, MAF, Agriculture, MAF, we call it MAF, for environmental safety or biodiversity safety assessment. And then for the food safety, we need to notify to the Ministry of Health and Welfare. And the both ministry made regulation rules based on the discussion. And then here you can see the rules by the Ministry of Agriculture, Forestry and Fisheries, MAF. And then to notify, we have to submit these data to the Japanese government, and which contain the fact that organisms do not possess a remnant of extracellularly processed nucleic acid or its replicate products as stipulated in the Cartagena Act. And actually, we prove our tomato by using a PCR and genomics and techniques to prove this this situation. And also the toximetrical species of the modified organism, taxonomical species, and the method of gene editing used for modification, and the modified gene and its function, and the change. So that's GAD3 is our target, and also the function is which is responsible for GABA biosynthesis, and the change of trait added by the modification and the increased GABA content, the trait we provided. And whether there is a change of trait other than those are described in E. And so then we grow our gene-edited tomato and collected the data for germination speed and fruit setting and the ripening, and also sugar content and so on. And the use of usage of organisms we are going to use for fresh and processing both. And the discussion on the possible influence of biological diversity when the organism is used. And we have to provide this information to the government before notification. And also for food safety, we also have to provide a similar data to the Ministry of Health and Labor and Welfare Ministry, and those and for the notification, we need to provide these information to the these ministries, such as maybe one and the two is the same with Ministry of Agriculture, and the second one, third one is a kind of off-target analysis, and also the allergen and also toxin, because the tomato contain natural toxin, which is a tomacin, and we have to measure the tomacin content at the red life stages. And so we have to provide those information to the Ministry of Health and Welfare. That means that we confirm the safety of our products. Then the consumer is satisfied this kind of data we already provided to the government. Okay, and then finally, we got some permission from the those ministry. We can use it for cultivation as just a normal tomato, and also we can eat it as a normal food, and then we can feed it if we feed our tomato to the animals. That we got 3 permission from the our government. And then another topic is maybe a labeling system, and then maybe it's okay. And so which is a labeling system is in charge by controlled by Consumer Affairs Agency in Japan, and their conclusion is they don't need to, according to their current conclusion, we don't need to make a labeling, because it is very difficult to distinguish the gene edited crop and non-gene edited crop. So that's why they are not they ask me to labeling. But we made a discussion with the consumers for a long time, maybe 3 years or 4 years. And but some of the consumers, they wanted to have information about our products. So then our university venture company decided to give a labeling. This is labels we are going to make it in our products, which is mentioned that this tomato has been improved by gene editing technologies, and notification to MAF and MHLW has been completed. So we are going to, because this is in the first or initial products, so that's why we decided. But after I don't know, several years, consumers say no, they don't need to labeling anymore. We are not going to labeling. But at this moment, we decided to make a labeling so far. And for the patent, as maybe this patent is already patenting system is already explained by the previous speaker. And then there is 3 basic patent related to CRISPR-Cas9 techniques. But fortunately, in the field of crop application, DuPont now is Corteva is accumulated these patent, and then just we need to discuss with Corteva how to use for the crop improvement. And then this company is discussed with Corteva. Then we got licenses from Corteva to use CRISPR-Cas9 technique in crop improvement. So now that's why we could start the commercialization in Japan. And finally, the crop, we need to get the public acceptance. And then I spend a lot of time to introduce what is gene editing techniques and plant breeding. And the point is that CRISPR-Cas9 is just a kind of a mutation breeding technique, which we can more precisely introduce a mutation in the target genes. Then to explain the here you can see the wild type of tomato, and this is a commercial cultivars. When we compare the genome of both wild type and the cultivars, we can find a huge number of mutation, which is created by naturally or artificially. So this kind of explanation is can provide a better understanding of what is gene editing technology to the consumers. And in addition to, I sometimes use this slide to explain what is the difference of gene editing and the GMO. Then gene editing crop is just we are not changing the gene numbers. Just we modify the present gene in the in tomato. But GMO, we add 1 or 2 or 3 genes. So that's why we have to evaluate the safetiness of this gene. So this is a differences in GE and the GMO. And then maybe most of the consumers can accept and understanding what is the difference of GMO or GE. Maybe this kind of activity is necessary a step to this communication with the consumer is necessary, very, very important step to introduce new techniques to the society. Okay, I think almost my time is finished. And then this is, I think, the last slide. So now new era of rapid and precise breeding of elite cultivar via gene editing techniques. And then now the automate breeding for sustainable food production has started. So thank you for your attention. This is maybe hot topics because this tomato has just commercialized in Japan, start commercialization in Japan. And thank you very much for your attention. I'm pleased to have your comment and your questions on our activities. Thank you.
Okay, thank you very much, Professor Ezra, for the interesting and well-delivered presentation. And now we are coming to the last presentation for this plenary session that will be presented by Dr. Mastur. Dr. Mastur finished his PhD program from the Tokyo University of Agriculture, Japan in 1999 with regulation. He is now the director of the Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development, ICA Biograd. And his title of presentation is Harnessing Plant Genetic Resources Through Modern Biotechnology for Self-Sufficient Food Production in Indonesia. Dr. Mastur, please, time is yours.
Thank you, Mr. Chairman. Assalamualaikum warahmatullahi wabarakatuh. Good evening, everybody. It is my pleasure to join this fruitful virtual conference to share about harnessing plant genetic resources through biotechnology for food security in Indonesia. Even though we have been combating COVID-19 pandemic, we hope still productive and safe from this virus. Here, the outline of this topic. You could see from this slide several issues. Challenge of food security, relevant biotechnology to harness plant genetic resources, status of Pgr in ERG gene bank, utilization of Pgr in Indonesia, super technical aspect, and future perspective and conclusion. Food is very important to feed the world. It is relevant with the statement of Chief Economics FAO, Maximo Terrero Callens, in the celebrating World Food FAO last year, 2020, that as country combat to coronavirus pandemic, they must also make every effort to keep their food supply chains smooth. In particularly, a country like Indonesia that experienced drought in some area due to climate change that will be affected on the plant production. In addition, FAO asks every country dealing with the spread of coronavirus to also maintain the smooth running of the food supply chain. These food supply chains involve complex interaction such as in the agriculture sector and other. Current major issue on PGR need attention. Over 1 billion people suffer from chronic hunger and malnutrition. Agriculture production and consumption in 2050 was projected to be 60% higher than 2005-2007. Due to this, the growing population should strive for food security based on sustainability in production, rational in consumption, as social, environmental, and health impact are very important. As consequence, agricultural production must increase by 70%. Therefore, PGR is essential for supporting food security and sustainable development, especially through breeding and biotechnology. PGR conservation and utilization are bases of agricultural production. PGR play an important role in enabling crops and livestock to withstand global climate change and to ensure food safety and security in the world. PGR is essential genetic material for broadening the genetic diversity to support breeding program. Clearly, genetic diversity is basis foundation required for variety selection development. Therefore, genetic erosion should be minimized since we will lose diversity, reducing opportunity to achieve goal in effective breeding and biotechnology. The PGR such as line races, local cultivar, breeding rights, new variety, and wild relative are essential to support food security through carbohydrate source such as cereal, tuber crop, beetroots, sagu, and other nutrients such as vegetable and fruits. Bioprospecting, PGR for producing phytopharmacy, cosmetic, biofuel, biomaterial for industry. Therefore, PGR are foundation for sustainable bioeconomy in the future. Principally, there are three functions of biotechnology related to PGR. There are conservation, genetic improvement, and propagation. With the advantage of biotechnology, we are able to do conservation using in vitro, cryopreservation, even conserve genetic material harboring interested gene. In vitro, marker-assisted selection, genomics, genetic engineering, genome editing, and other modern biotechnology are attributable for plant genetic improvement. Unlike selection by farmers who address on local varieties to complement conventional breeding, biotechnology has ability to generate improved variety with their right IP protection, including breeder right or plant variety protection patents, and also other intellectual property right. In the next few slides are about challenge for food security. PGR need to be constantly enriched, conserved, maintained, and documented for direct utilization. Different strategy and method for conservation has been made, such as ex situ, in situ, on-farm conservation. However, these activities are prone to the problem of genetic erosion activated as a natural process by artificial intervention or by both. The genetic erosion is commonly known to be resulted from modern intensive agriculture activity, such as a replacement of local variety by modern varieties, habitat destruction, over-exploitation of species, population pressure, and environmental degradation. This factor make it as a narrow genetic base, genetic variability of species, lowering ability to adapt to change environmental condition and loss of gene for nutritional food traits. Conservation of wild relative habitats remain undervalued compared with alternative use, such as a clearing for agricultural and urban use. Furthermore, agriculture and food production today is facing with many challenges, such as climate change, land degradation, increasing human population, and biodiversity loss. Food security is determined by ability of household or region to maintain its own food requirement, which can be measured at different levels. To supply daily food and nutrition capacity for production distribution, and accessibility are important factors. Therefore, a rural region can increase the stability of food system by building its own food self-sufficiency through improving productivity, quality, and efficiency of farming system. Invention and innovation are key strategy to achieve food security. At this topic, I will address an issue on relevant biotechnology to harness Pgr. Pgr including relative species, linearizes, local varieties, breeding lines, and improved variety are very important as an input into selection process through plant breeding to develop new and improved varieties. The Pgr that need to sustainable utilization to achieve food security through application of in vitro manipulation, cell fusion, transfection, transformation, genome editing, and DNA and RNA in complement to previous generation of biotechnology to support breeding and regeneration of plants. Three types of genomic contribute to crop improvement that are: first, comparative genomic, referring to comparison of genes from different genomes to related functional and evolutionary relationship. Second, functional genomic, which involve studying functioning or regulation of gene expression. Third, structural genomic, which determine complete sequence and gene map. Here are some examples of the application of genomic on crop improvement, such as genomic size, gene number, gene mapping and sequencing, evolution of crop plants, gene cloning, identification of DNA marker that can be developed as a marker-assisted selection, transgenic breeding, linkage map, and QTL mapping. This improve on new varieties and related technology could be beneficial not only to plant production, food safety, human health, but also better environment. Focusing to ICGRB agriculture biotechnology research related to Pgr, we manage the national gene bank and local seed bank across the country to support research activity like bioprospecting, genomic and genetic engineering, including genome editing and conventional breeding, which are complementary to each other. Since genomic sequencing has become easier and cheaper for developing countries, we created a large national genomic database named PGPI. Now we manage all this GR research system based on our IP management of genetic resources. Let me introduce status of Pgr and EARD gene bank. Indonesia is a biological center of origin, rice javanica, banana, mango, pepper, coconut, sugarcane, sugar palm, taru, mangosteen, sandalwood, clove, and bamboo, and many several plants. And Chinese Indonesian Center for Secondary Diversity, some plants such as cassava, beans, sweet potato, coffee, and tea. Such high diversity of ICGRB in Indonesia need to be maintained in ex situ conservation and as a key element to achieve the Feed the World 2045. Therefore, in national level, we have ICGRB Gene Bank for Pgr under coordination of ICGRB. You could visit this link and explore what you need. In Indonesia, PGRFA is sponsored through several programs under Ministry of Agriculture. Under the Indonesian Agency for Agricultural Research and Development, ICGRB, PGRFA is sponsored in some institutes. Some perennial PGRFA are sponsored on farm under monitoring of Assessment Institute for Agricultural Technology, AEAT. Ex situ conservation is performed by Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development, ICGRB, and some crop-based research center along with their mandate. The EARD Gene Bank is facilitated with gene bank laboratory, field gene bank, in vitro laboratory, computer facility. In this gene bank, total 2,876 PGRFA accession are conserved in seed bank, field gene bank, and in vitro conservation facility. Here, I also would like to address the utilization of Pgr in Indonesia. Program on Pgrfa has been implemented at the institute under ICGRB, working on community in coordination with the National Center for Genetic Resources. The program consisting activity related to conservation and utilization of Pgra through breeding program. Importantly, we also reported part of our Pgrfa have been adequately characterized and evaluated. Information system on germplasm characterization have been also established in several institutes. Crop improvement program, which consists of conventional breeding, irradiation-induced mutation, and biotechnology, have been applied on various crop and trees, such as food crops, horticulture, and estate crop. Left figure, content phase in Indonesia for utilization of plant genetic resources. The first, lack of characterization and evaluation. The second, lack of documentation, lack of capacity, weak policy implementation, lack of integration between conservation and utilization program, lack of coordination among researcher, breeder, gene bank manager, and farmers. The right figure, to enhance the use of plant genetic resources in Indonesia, the following activity has been undertaken. Strengthen capacity and improve training in plant breeding, increase collaboration, more attention to using developing underutilized species, and implemented participatory approach to plant breeding, explore marketing opportunities, promoted the use land races, farmers varieties, local varieties, and improve the regulatory and policy framework. Those are current priority for Indonesia and our needs to implement them. Biotechnology-based breeding conducted in Indonesia was implemented by application in vitro culture technique using in vitro selection, culture, embryo rescue, protoplast fusion, molecular breeding using molecular marker, and genetic engineering. This breeding strategy aimed to the genetic improvement through some important traits such as resistance to pests and disease, increased tolerance to drought, phosphate deficiency, aluminum toxicity, iron toxicity, increased sugar content, increased oleic acid, delay ripening, free amylose, and different crops such as rice, soybean, sorghum, citrus, chili pepper, potato, and tomato. A great achievement in the IP management for genetic resources. Related innovation is the liaison agreement of Ministry of Agriculture, consisting of plant variety protection, patent, trade secret, and other branches of IP, have been almost double since 2016 up to 2020, with a total number 363 license agreement. Some of produced technology included this developed from genomics. Reportedly, around 114 private sector have adopted 139 ERD technology with specific agreement for benefit sharing. Here is supporting legal aspect on national and international level. To secure legal and technology ecosystem for genetic information, IP-APS framework on Pgr data, including genomic information, digital sequence information should be addressed. There are some international policy in the IP and IPS and EPS of genome information. There are relevant CBD, FAO, ICGRB, ICGRB, WIPO, WHO, VAP framework, UNCLOS, and OECD process. The genomic sequence, and other digital sequence information resulting from the omic disciplines linked with the genome editing to other technology utilized in synthetic biology should be solved. How the beneficiary should be. All of the supporting information are very important, especially in breeding, as improve and developing new variety are the target for food production. In favor to harness PGR-FA, a number of governmental policy framework and law support national PGR program of conservation and utilization either on non-genetically modified crops or genetic modified crop. Notably, Indonesia ratified the International Treaty on Plant Genetic Resources for Food and Agriculture and Nagoya Protocol. The access and exchange of PGR-FA included in Annex 1 of the ICGRB volume multilateral system, the remaining of plant species access is in accordance with the bilateral agreement of Nagoya Protocol under Convention on Biodiversity, along with relevant national legislation. Since research in IERD produce a lot of genomic sequence and molecular biology analysis, this PGR data management need to be addressed for their protection. IP data domains in IERD hopefully will be managed in different domains, such as a public domain, substance domain, and internal-only domain. IERD PGR research is now used as a model using WIPO toolkit management model. We also link with ENE system as an example of IERD data test, which could be important for future collaboration for food self-sufficiency. The future perspective included 4.0 Industrial Revolution to support PGR management. Biotechnological approach such as molecular marker, genome editing, genomics, and other omics will be incorporated into national breeding strategy and national agricultural development to achieve self-sufficient production in Indonesia. To more intensive culture, IP and APS management on PGR and PGR data. Supporting legal aspect and well-implemented regulation in support of biotechnological research and development. Finally, I conclude that the potential of highly diverse plant genetic resources in Indonesia need to be harnessed for plant development and sustainable agriculture. Food security in Indonesia is challenged, and biotechnology research will be essential to solve the problem. Advanced technology of genomics and other omics, genome editing, and other modern biotechnology approach are important to bridge the gap of genotype, phenotype, genotype environment for food production. The IERD mandate for plant genetic research for food and agriculture, PGR-FA, the scientist has developed many products, releasing new varieties and technology with vision of the intellectual property-based innovation system for GR. Secure legal and technologies ecosystem are important PGR and genetic information. That's all of my presentation. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Dr. Master, for your interesting presentation. Before we start with the discussion session, I would like to invite the invited speaker to be ready for this session. Okay, Dr. Michael Thompson is already there, and Dr. Ezra. Thanks to all the invited speaker for the interesting and well delivered presentation. We now have the discussion session, and please raise your hand if you have any question or comment, and please mention your name, your institution, and to whom your question or comment will be addressed. If there is no direct question, I will directly go to the question from the chat box. Please just raise your hand. Ada enggak? Enggak ada. Enggak ada. Okay. So far we have some question for Dr. Thompson and Dr. Azura. I think we will try to divide this question for the first part of the session. I will give two questions to Dr. Michael Thompson first, and then two to Dr. Azura. Okay. The first question for Dr. Mike Thompson is from Mr. Gufroni, and the question is how we can determine if the result is a non-GMO and what institution can provide non-GMO certificate for this product. And the second question for Dr. Michael Thompson is from Dani Setiawan from ICGRB. And the question is, if money is no object, is it better to deliver CRISPR as nucleic acid or ribonucleoprotein complex? And then for Dr. Azura, the first question is from Lina Herlina ICGRB. When applying gene editing to a plant species, will the same target genes be able to directly apply to other species, or must it always be started by providing complete genome data first from the species? And the second question for Dr. Azura is from Bu Bahagiawati from ICGRB. The question is, was it difficult to obtain patent-free or license for getting commercial permit of GABA tomatoes? How long did it take? Did it also took some cost for doing that? Okay, you both have two questions for the first part of this discussion section. Please, Dr. Thompson, to give your response for these two questions.
Okay. Yes, thank you very much for the questions. And so for the first question, how do we determine if the result is non-GMO? I think, I mean, the key is that there can be no foreign DNA sequence integrated in the genome, right? And so I think when you do that practically, you can design PCR primers for each of the foreign DNA segments. So Cas9, also selectable marker. If you use hygromycin, you could just design PCR primers for each of those for anything that's within that foreign DNA sequence. And then if you, for instance, after several generations, if you can get rid of the foreign integration and then use that PCR to determine that there's no remaining foreign DNA in the genome. And so I think that's really the way it's done. And I will mention in the US, again, so USDA regulates if it's GMO or not, but they actually currently really allow self-determination. And so it's up to the producer to determine whether it's essentially has no foreign DNA in it. But there is a nonprofit organization called the Non-Gmo Project. And if you see, at least in the US, they have a little symbol they put on the food that will say non-GMO. And so you can, people also can apply and they do independent testing to determine if it's non-GMO as well. So, and for the second question, which one's better, nucleic acid or the RNP approach? I think many people, really would prefer to use RNPs just because then there's no chance that you'll have maybe, for instance, that DNA integration. Sometimes if you use GeneGAN, you'll have like small fragments of DNA that may be integrated in multiple copies, and it can be a little difficult to clean that up. And so RNPs actually, since there's no DNA involved, there'd be no chance for that DNA integration. But at the same time, they're much more difficult to use, and it's very hard really to optimize RNP editing right now. They have still very low frequency of mutations. And so it would be a lot more work. And so I think, practically speaking, most people still use agrobacterium or GeneGAN just because it's so much easier to use. But theoretically, I think RNPs may be preferred just because you have that. There's no chance of that DNA integration. Thank you.
Thank you, Dr. Thompson, for your response. And please, Dr. Ejura, to respond the two questions.
Okay, so thank you. Okay, thank you for the question. And for the first question, which is the okay, so about the target gene of the gene editing. Now, if you're targeting a kind of a general trait in the plant development, so we can maybe target the same gene that crop species you are interested in. Actually, in case of GABA example, GABA is a basic or primary a compound contained in all kinds of organisms, and then in plant, the biosensitive or metabolic pathway is almost the same. So then you can target the same genes with us, with tomato, and if you're interested in other fruits, crops, fruit species, and then you can target the same genes. But if you are interested in some specific trait in some crop, maybe you have to identify the target gene. So maybe that is a general point for the targeting genes. That is an answer to the first question. And the second question that you are asking the how long will it take to develop the crops, and how long how much is the cost of the patent? Okay, so actually in our gene edited tomato, we spent just less than 2 years to once we obtained the parental line for elite F1 hybrid cultivar, then we started the genome editing. Then less than 2 years, we can improve the trait to be a high GABA content. But this is also up to a plant species, crop species. And if you are targeting the crop species, which is very difficult to genetic transformation, and that will take a long time. But if you are interested targeting crops like tomato and lettuce and some other vegetable crop, which is easy for genetic engineering, it takes a short time to improve. And about the patent, so this is maybe I think this is not a general cost. It's depending on the how to use and how to who is doing this going to using these techniques. And then, so if you are interested in about the patent fee and something, you need to or developer have to talk with the patent holder in case of like a crop application. Maybe developer have to discuss with Koteva companies, and then they will give you a more precise idea how much cost of the patent fee. That's is it? Yeah, that is my question. I can say, but yeah, so far. But actually, the startup company, university startup company, which I introduced, Salinity seed company. This is a very small seed company. So then, a patent is acceptable price for like application. Maybe we don't need to so afraid about the patent, because if the developer cannot get any income. So maybe a patent holder cannot get any advantages. So maybe it's a kind of a discussion with the developer and the patent holder companies, holding companies. And then I think that my suggestion is that this Corteva is quite, if the developer is going to apply this new technique for increasing sustainability, food sustainability in the developing countries. So maybe they can maybe freely use these techniques. It's depending on how this technique has been used. I heard that the cassava is Corteva is supporting cassava development because of cassava is an important crop in the tropical region all over the world. So if you are wanted to interested in use these techniques, maybe I recommend you to talk with the people in the Corteva. And the Corteva is maybe an office even in Indonesia, I think. So then you please start a discussion with Corteva companies in Indonesia. That's my suggestion. That's
okay.
all.
Okay, thank you for your response, Dr. Azura. And now we go to the next segment of this discussion. Again, to question for each speaker. For Dr. Thompson from Sri Kurniati, Ikabayograd. This is the first question. If we work on hexaploid plants, how possibility to use this system to knock out the gene target? And the second question from Admitri, Ikabayograd. What is the difference between a gene drive and genome editing? And then for two questions for Dr. Azura, from Niputu Megayani, PT Prima Instrument Analytica. When we develop the genetic modification in tomato, are there any effects in the flexibility in growing that plant in the field? For example, after genome editing, the mutant tomato must grow in greenhouse with maintain stable temperature, or it more easy plant in the field. And the second question from Dani Satyawan, Ikabayograd, to Dr. Azura. You have a lot of interesting tomato mutants. Did you have a certain goal? For example, to find long shelf life mutant when screening the mutant, or did you try to observe everything and keep the interesting one? Okay, please, Dr. Thompson, respond that question.
Okay. Yes, thank you for the question. So for the first, if we work on hexaploid plants, it is certainly possible. There are some publications from wheat for gene editing, but I would say it's much more difficult because, as you know, wheat has like A, B, D genomes, right? So you'd have to target the gene in each of the three genomes. And practically that means you can design, for instance, 3 different guide RNAs for specific polymorphisms in each of the genomes, or potentially you can have a single guide RNA that targets a conserved region that actually will hit all 3 genomes. But in both cases, you need larger numbers of progeny. So when you do the transformation, you'll need a larger population because the chance is less that you'll have all 3 genomes, knocked out at the same time. And so it is just more work, I think. But there are some publications in wheat, and so it is possible. And for the second question, the difference between a gene drive and genome editing, I would say, genome editing is a process. It's a technology that has many applications, and just one of those applications is gene drives. And so, at Texas A&M, there's actually a group working in mosquitoes, and they're actually looking at how can we use gene drives in mosquitoes, essentially to take the population and modify the frequency of the progeny using the gene drive, and they can use genome editing as the process for that. But the gene drive is what essentially changes the frequency of the progeny in the next generation. And theoretically they could actually eliminate all the mosquitoes in the world using that technology. But of course, it's very controversial because people worry that there may be some unintended effects if you don't have any more mosquitoes, right? And so I think that's why gene drive is so controversial, because it's a very powerful technique. But I haven't seen it used in plants very much. So I think it's not yet, I think, very common, and people are being very careful because it is such a powerful technique. Thanks.
Okay, thank you, Dr. Thompson, for your response. And please, Dr. Izura, to respond the two questions for you.
Okay, thank you very much. For the first question, the question is that is there any effect on the other developmental trait when we engineer that GABA accumulation in tomato? And okay, so in this case, we only engineer the GABA accumulation in the fruits because we targeted only a fruit-specific expressing genes. So then in our tomato, we can only increase the GABA content in the fruits. And other part of the root and the leaves, and there is no changes of GABA accumulation. So that's a growth or growth of this high GABA tomato we developed is almost the same with their original line. So then and our tomato is we can grow it in an open field, and then so that's a growth performance is almost the same. And it's depending on how you use this gene editing technique and how you are going to targeting the gene. So if you are carefully choose the targeting genes, so then you can only change the trait of targets. So that is a question to a first answer to a first question. And the second question is about the mutant correction. As I explained, we have a huge number of mutant correction based on a microtome. It's a model tomato species, but we can cross it with a normal tomato. Once then that means that once we found a useful mutant, then we can introduce a mutation in the cultivating tomato. And so question is interesting in the shelf life of tomato because of the maybe your maybe shelf life of the tomato is some problem in Indonesia. So that's actually we already screened the several mutants lines showing extended shelf life. And then I have a lot of alumni from my laboratory in Indonesia. They are now teaching in the Pajajaran University in Indonesia. And so then maybe my alumni is three alumni who got a PhD in my laboratory, and they are teaching in that university. And we already collaborating with the teams to introduce a long shelf life trait, which we isolated from our mutant correction. And they are now the breeding to extend the shelf life into Indonesian tomato is ongoing in the Pajajaran University. So that's the question are interested in the topics, and you can discuss with the people in the Pajajaran University. And they are already conducting such a research from maybe four years ago already. Yeah, thank you for the question.
Okay, thank you, Dr. Izura. We still have some question for you both. For Dr. Thompson, the question is from Bu Bahagiawati, Eka Biograd. The question is, how many products of genome editing have been approved for commercial purpose in the USA? And the second one is, what requirement that we need to fulfill in ensuring products derived from genome editing technology is not a GMO? And one question for Dr. Izura is from Ibu Eni Daryanti Ikabayograd. The question is, does genome editing crop categorized as GMO is in Japan? How does the regulation for these products in Japan? Okay, please, Dr. Thompson, to respond this both question for you.
Okay, yeah, thank you for the question. And so, as far as I know, there's really no CRISPR food crop that's commercialized in the US at the moment. But I do know that there is one gene-edited soybean that used the TALEN technology. And so that was the previous genome editing technology. There's a company called Calyx out of Minnesota. So Dan Voitas is kind of leading that. And they have a high oleic acid soybean oil using the TALEN genome editing technology. And that's already commercialized and actually being used in restaurants for frying, because high oleic acid is a healthier oil for frying. And so that soybean oil actually has been commercialized. So as far as I know, that was the first genome edited crop, but that wasn't using CRISPR, that was using the TALEN or TALEN technology. But I think there are several that are really probably already approved, but just not yet on the market. I heard that there's a waxy corn that Corteva was working on, and there's a report of the brown mushrooms out of Penn State. So there's quite a few products using CRISPR, but as far as I know, they're not yet on the market. For the second question, essentially, so what are the requirements to ensure it's not GMO? So I mentioned the no foreign DNA sequence. And again, at least in the US, if it's USDA approved, you also have to have one of the 3 exemptions. And so that's like a simple deletion or single base substitution or a gene insertion from the same gene pool. So as long as there's no foreign DNA sequence and you fall under one of those exemptions, essentially then from the regulatory perspective, it's non-GMO. But again, that could be, you can have different definitions in different countries. So it really depends on the country that you're in. And so there's the regulatory definition, and then maybe even, practically speaking, like the consumer acceptance definition. So many people will still combine, gene editing as GMO, but at least in the US, it can be non-GMO. So, thank you.
Can I answer Can I answer now? Okay, so
sorry, I have to unmute this.
Okay, so
Okay.
my question is that it's a gene editing crop is a GMO in Japan or not? And the definition is the same in the US. And as I mentioned, that 3 types of gene editing technology so far. We have SDN1, SDN2, and SDN3. And if we produce the crops by SDN1, and then we can prove that the absence of a foreign gene in the crops, that will be considered to be a non-GM in Japan. That is the same regulation in the US. And yeah, so that's and I think at the end of the April 30, the European government also released a new statement about the how to handle a gene-edited crop. And I think they are stating that mutants created by traditional mutation breeding and also mutants created by gene editing, there is no clear differences. And so that they say that to distinguish those 2 products is not realistic. So that's the new statement just released for 3 weeks ago in the EU government. So that's why I think that maybe this rule is going to be same in all over the world. But in Japan, as I mentioned, that the same in US, we are the crop it does not have any foreign genes and produced by SDN1 technology. That's considered to be a non-GM in Japan. Thank you.
Okay, thank you, Dr. Ijira, for your response to the question. And we still have 2 questions, and these 2 questions are from Dr. Thompson. The first one is from Bu Puji Lestari from Indonesian Center for Rice Research. The question is, how is the main difference between CRISPR and transgenic technology, and how fast are the new plants produced by CRISPR? And The second question is from Pak Sabtowo from Ika Biograd. The question is, what is the current status of regulation for plant products produced from CRISPR technology? Okay, please, Dr. Thompson, to respond these two questions.
Yeah, so thank you again for the questions. And so, I mean, for me, the main difference between CRISPR and transgenic technology really is that, you know, when you use a CRISPR insertion, so like Cas9 guide RNA, the mutation is done essentially somewhere else in the genome. And so it's, you know, usually on a different chromosome. Whereas if you're inserting a gene with transgenic technology, normally you need that gene, you know, that insertion to remain in your genome to have the desired effect. Whereas with CRISPR, you can get rid of the insertion. So the Cas9, you can remove it in the next generation, and your mutation will still be there. So it's essentially, it's independent. The insertion is independent from your phenotypic effect, right? So that's why CRISPR then, you can, even if it's transgenic, you know, one generation, the next generation, you get rid of the foreign DNA insertion, and then it would be essentially non-transgenic after that, but it still has your desired mutation. Whereas most transgenic technologies, it's very hard to get rid of the foreign DNA because that's what's, you know, causing the effect that you need. So that's why, that's the main difference. But at least, you know, how fast is it? It's just still follows the traditional transformation techniques for the most part. So that differs for each crop. But often rice, you know, you can do it in 4 or 5 months. It depends on the, again, on the genotype or the cultivar. But it's still relatively fast. I mean, for most crops, you know, certainly less than a year, you should be able to get a product out of it, at least for the first generation. And then as far as, you know, the current status of regulation, again, so the, you know, I mentioned USDA APHIS, which is the agency that deals with plant pests. And so they regulate if you can have a transgenic crop planted in the field or if you can transport it. And then in the US, there's also the FDA, is the Food and Drug Administration, reviews food safety. And then the EPA, the Environmental Protection Agency, reviews if there's any pesticides or if a plant produces a pesticide or herbicide, that's also reviewed. And so I think, at least for APHIS, it's, you know, gene editing falls under those exemptions. It's okay to plant, but then you still have to go under review of the FDA. And that's just case by case. Essentially, all new food crop varieties have to go under this kind of review. And so that's, again, just kind of case by case. But I think, you know, in the future, this will be fairly quick approval process. And so it shouldn't be, I mean, it should be much easier than transgenic crops in the past. Thanks.
Okay, thank you, Dr. Thompson, for your response. If anybody still has any question, please raise your hand. We still have about 5 minutes before we finish with this session. Siapa? Ada? Just wait for a second. We are still checking if there is any question. Okay. This one question for both of you from Enida Rianti, Ika Biograd. The question is for GE product, whether categorized as GMO or non-GMO, but you always have to give the proven analysis to the regulator. so which office will evaluate the document and make the decision? Maybe there is a little difference between US or Japan. So please respond, Dr. Thompson and later Dr. Ezura. Thank you.
Yeah. So as far as the US is concerned, it's from what I understand from the secure rule, which was recently published, is that it's really on the the innovators or those who produce the gene edited product to check to make sure there's no foreign DNA in those products. And again, it's the initial agency is the USDA APHIS, but it's largely self-determined. I mean, they'll run the analysis and then they can essentially prepare a report that says, that there's, essentially no foreign DNA and then it's non-GMO. And so there's very little regulatory burden. I think it's actually, once you've determined that, it's quite easy at that point, at least in the US. And so there's, for instance, there's not as much, I would say, kind of evidence you have to give as with the transgenic regulatory process. So it's a lot easier, I would say.
Okay, so in Japan, so as I explained that we have two ministries in charge of this decision or evaluation and decision. One is the Ministry of Agriculture and Fishery and Forestry, which is and another ministry for food safety assessment. It's a Ministry of Health and Welfare, and those are two ministries responsible for the evaluation, the decision of these products. And each ministry, they have a committee to the which include a different field of specialists, and so those committee discuss the data and some information, and then finally those two ministries decide or make a decision on this crop, gene edited crop.
It's already done. Your response? Okay, thank you. Okay, ladies and gentlemen, oh, okay. There is another question from Pak Guproni. Please, Pak Guproni.
Yes, thank you. Hello?
Yeah.
Oh, yeah. I have a question for Professor Hirohi Ezura. My question is the gene editing product considered as organic product? And the second question is if this product is safe for this or meet the sustainability goals?
Yeah.
Thank you.
Okay, I think that first question that still we have no decision about which included in organic product or not. Okay. The government is discussing. And so your second question is that can I repeat could you repeat your second question again?
Yes. Is the gene editing product consider meet the sustainability goals?
Mm-hmm. Okay, so that's a very, very important question, and okay, so it's depending on what kind of trait we are going to improve by these techniques. For instance, we can improve the a heat tolerant, so then that will be directly contribute to the sustainable, and also I think that promoting the health through food or meal that is one phase of the sustainable goal. So that's if we can target that's improving such a trait, that will meet the sustainable goal.
Okay,
thank you.
Pak Guproni, it answer your question?
Yes, I think it's I get the information. Thank you.
Thank you.
Okay, Ladies and gentlemen, finally, we come to the end part of a plenary session. I will not conclude anything. It's already clear, I think. I would like to thank again to the invited speaker for the interesting and well-delivered presentation, and also to all participants for active participation. Hopefully, the presentation will be beneficial for everybody. Let us give a round of applause to the invited speakers. And I will give this floor back to the organizer for the next agenda of the conference. Thank you for your attention.
Thank you.
Thank you, Dr. Yunus, and the valuable invited speakers. I hope everyone had a productive discussion today. Next, we will have a 1-hour lunch break. This break will be filled by our sponsors. PT Crop Life will presenting some materials, then PT ITS Science Indonesia, PT Fajar Mas Murni, and PT Prima Instrument Analytica. After that, please all participants can return at 12:30 Western Indonesian times, since we will have a quiz and the winners will get some attractive goodie bag from us and sponsors. The quiz will be hosted by Dr. Hakim Kurniawan. After the quiz, we will then start the parallel sessions at 1 p.m. Western Indonesian times. We will move you to the breakout rooms. We have 3 rooms today: Room 1 Aglonema, Room 2 C Palandra, and Room 3 Citrus. But if you want to watch some presentation in another room, just message the organizer using private chat and we will move you to that room. Please, speakers, pay attention to your schedule in program book. Okay, I would like to pass it to Mr. Alex from PT Crop Life and then continue to Dr. Kurniawan. Okay, all participants enjoy your lunch while watching nice presentation and video. I will see you again after the quiz. Bye.
The keyboard.it link as showing in the Google search result. After you successfully get into the Kahoot, then enter the PIN as we will show later what is the secret PIN. And then you can enter your unique name, your unique nickname. And now you are ready to take the quiz. When the question is previewed in the screen, you can select the symbol that corresponds to your correct answer. In this quiz, there will be 6 questions. It is kind of multiple choice questions. Please pay attention that you only have about 20 seconds to answer each question. Otherwise, you will miss your chance to answer these questions. When you finish the quiz, we will select the first 3 participants who won the highest score. An interesting prize gift is available for these 3 winners. Okay, are you ready now? Let's start the quiz. Okay, now we have 30 participants that already joined the quiz. Shall we start now? Are you ready? Okay, this is the first questions. Who is the Indonesian Minister of Agriculture? Please select your correct answer. Amran Sulaiman, Syahrul Yassin Limpo, Suswono, or Anton Apriontono. 18 answers, 9 answers. Okay, the correct answer should be Syahrul Yassin Limpo. there are 15 participants that taken the correct answer. And you can see here the highest score is taken by Mrs. Rini. Next questions. In what year was ICGRB, the first ICGRB held? In 2017, 2018, 2019, or 2020? The correct answer should be 2018, and there are 9 participants that taken the correct answers. And now, Mrs. Rini still take the winners for the quiz. That's nice. Okay, now for the next question. Is the sticker of genome editing of tomato? Please select the correct answer. 22 answer, 23. 26. The correct answer should be Hiroshi Ezura, and there are 20 participants that taken the correct answer. And now, the first position is taken by Mrs. Rere. Congratulations. Now we go to the next questions. The following are located areas of the food estate program, except Java, Sumatera, Central Kalimantan, or East Nusa Tenggara. The correct answer should be West Java. There are 11 participants that taken the correct answers. And now, who taken the first one? It's taken again by Mrs. Rini. Okay, congratulations. Yeah. Let's go to the next questions. The following are the new superior varieties released by Ika Baikurat, except Yusoi, Nakula Sadewa, Yubestari, or Impari Blast. 23 answer, 24. Okay. The correct answer should be Nakula Sadewa, and there are 12 participants that taken the correct answers. And now, who took the first positions? Mrs. Rere again. The last questions. What is the ICGRB theme this year? Please choose the correct answer. This is the last questions. The correct answer should be the last one, harnessing technology for conservation and sustainable use of genetic resources. And there are 14 participants that taken the correct answers. And now, who is the winners? The third one is winners. Second one is AR, and the first one is Mrs. Rere. So this is the 3 person that taken as the winner of this quiz. Congratulations for the 3 winners. Please contact the organizer as soon as possible to claim your prize. Thank you for your participation, and see you on the next second ICGRB quiz for tomorrow. I am Hakim Kurniawan. Bye.
Thank you, Dr. Kurniawan. Good afternoon for all, ladies and gentlemen. I hope you enjoy the quiz, and congratulations to the winner. For the winners, please chat the organizer to give your name, address, and contact number. Okay, for the parallel sessions, We will divide you into 3 rooms. Are aglonema, Butse Palandra, and Citrus. If you want to go to another room, send a private chat to the organizer and we will move you to that room. Today's parallel session will be led by Dr. Sri Kurniati in the aglonema class, Dr. Alina Ahdia in Buse Palandra class, and Dr. Levri Nur in the Citrus class. So just sit and relax while we move you to your respecting room. So I would like to hand over the floor to the 3 of moderators. And we will begin if all already set. See you for tomorrow and enjoy the parallel sessions. Goodbye, stay healthy. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Good morning for everyone. We warmly welcome you to the second International Conference on Genetic Resources and Biotechnology. We hope everyone in good and healthy condition. Also, high optimism that conference will run smoothly, better than before. I will mention again, this international conference is made possible through the collaboration between the Indonesian Agency for Agricultural Research and Development and the Indonesian Biotechnology Consortium, also fully supported by our partnering companies. Ladies and gentlemen, due to cancellation of one invited speaker, we reschedule the parallel session. The parallel session will start at 11 Western Indonesian time. Now we enter the plenary session. It will be moderated by Eni Idarianti, PhD. So I would like to hand over the floor to Mrs. Rianti, and I will see you again at the end of the first plenary session.
Thank you very much, Madam MC. Our distinguished guests, the DG of ICGRB, Director of ICGRB, invited speakers, all participants, ladies and gentlemen, good morning and Assalamualaikum warahmatullahi wabarakatuh. Welcome back to the second International Conference on Genetic Resources and Biotechnology, Day 2. We hope the second day is as interesting and enthusiastic as day as yesterday. My name is Eni Idarianti from ICGRB. I will be your moderator in this plenary session. For the second day, we're supposed to have 3 speakers. The first one is Dr. Ines Atmo Sukarto from Australian University. And second one is Professor Bambang S. Purwoko from Sunil R. from National Bureau of Plant Genetic Resources, India. However, we regret to inform you, as the MC mentioned before, that Dr. Ines has very urgent reason for her presentation in very short notice, and we cannot provide replacement for her slot in very short time. So we apologize for that on behalf of the committee. So we really have 2 speakers today. The first speaker will be Professor Woko from IPB University, and the second speaker will be Dr. Sudhir Arsak from National Bureau of Plant Genetic Resources, India. So they gave me new allocation time from 9 to 10:30 Western Indonesian time, but there is plenty of time for 2 speakers, I think. And each speaker will deliver their presentation, and after that we have panel discussion. For all participants, if you have any questions, you can raise hand by pressing hand logo below your computer screen, or you can write the question in the chat box. Mention the name of the speaker you address, you can recognize you. So at least for the first speaker, We have Professor Bambang Purwoko. I can see from the participant. Professor Purwoko already available. So of all, I want to introduce you Professor Bambang Purwoko CV. Professor Bambang was graduated for his doctorate degree from Cornell University in the US in 1992 and achieved his professorship in 2005 with a teaching experience from 1984 up to now. Professor Bambang research focused on the interculture of rice and horticulture crops to accelerate breeding program with emphasis in the tolerance to abiotic stresses, tissue culture, and biotechnology of rice and horticulture crops. You can read the paper published, research, and academic activity in the program book. Today, Professor Bambang will deliver his presentation with title Haploid Technology to Accelerate Breeding Program. So, Professor Bambang Purwoko, time is yours.
Thank you, Ibu Moderator. Good morning, ladies and gentlemen. First of all, I would like to thank the organizing committee for inviting me to this respected conference. I am essentially the same meaning, haploid technology to accelerate plant breeding program. Oh, sorry. I hope can it be seen? So
yes, Pak.
To all of the participants.
I can see your presentation.
Okay. Okay, thank you. So the outline of the presentation is as follows. First of all, of course, the introduction and the occurrence of haploid, supporting technology for double haploid production, and then characterization of the regenerant, how double haploid accelerated plant breeding, and then methods and factors influencing the double haploid production. Then some application of haploid technique for crop improvement, and last is an example of successful haploid breeding production in several important crops. Plant breeders have always been interested in shortening the period required for inbreeding. Previously, breeders used single seed descent breeding method. This is the classic procedure of having a single seed from each plant, bulking the individual seeds, and planting out the next generation. It is a method of inbreeding, segregating population that is quite conducive to environment that are not typical. However, with the advancement on plant and tissue culture, now double haploid technology is one of the fastest tools available to develop potentially a large number of inbred lines in shorter times. Perhaps the most thorough statistical analysis of the field performance of agriculture-derived double haploids was by Cortes in 1993 and Rice, who compared the mean genetic variance, and kurtosis of three double haploid populations to their respective single seed descent population. He found that single seed descent and agriculture-derived double haploid breeding should be equally effective for developing new lines. Thus, both double haploid and single seed descent breedings attempt to reduce the time required for inbreeding by rapidly advancing generation without breeder or natural selection. Haploids can be induced either naturally or induced by various methods using female and male parts of the plant. So naturally, we can use first parthenogenesis. For example, is semigamy. It is an abnormal type of fertilization whereby either reduced or unreduced male and female gametes participate in embryo formation, but fertilization does not occur. Parthenogenesis resulted in maternal haploid. Then second, the apogamy. For example, is polyembryony. This is the production of 2 or more embryos in one seed, owing either to the existence and fertilization of more than one embryonic sac or to the origination of embryos outside of the embryo sac. The induced one, there are several. The first is androgenesis. Male parthenogenesis, in which the embryo contains only parental chromosome, owing to the value of the egg nucleus to participate in fertilization or the regeneration of whole plant from sexual male cell culture, in this case anther or isolated immature pollen. Second is gynogenesis. This is spontaneous induced female parthenogenesis, in which the embryo contains only maternal chromosome, owing to the value of the sperm cell to fuse with the egg nucleus. Then the chromosome elimination. It can be interspecific hybridization, which is a crossing of 2 species from the same genus. This allows the exploitation of useful genes from wild and improved species for the benefit of the cultivated species, such as in the bulbosum method from barley. The development of a haploid embryo by fertilizing an ovule of Hordeum vulgare with pollen of Hordeum bulbosum, followed by the subsequent elimination of the chromosome of the pollen source, allows us to obtain double haploid from Hordeum vulgare. In intergeneric hybridization, it's a cross between plants of 2 different genera in the same family. This crossing represents an opportunity to combine genome from distinctly different plants and to integrate traits not found in the main genus of interest, such as in wheat cross to maize, to obtain double haploid wheat. Then intraspecific crossing. The development of a haploid embryo by fertilizing an ovule with pollen of the same species and the subsequent elimination of the chromosome of the pollen. The pollen source known as haploid inducer. For example, maize A cross to maize B, which is a haploid inducer acts as pollen source to obtain double haploid from maize A. Then pollination of irradiated pollen, which is development of haploid embryo by fertilizing an ovule with irradiated pollen that nevertheless is capable of introducing cellular division in the ovule and the development of the embryo. What are supporting technology needed in double haploid production? First, technique to make haploid tissues or plants from heterozygous parents. At the haploid level, every gene is hemizygous. Haploid plants are typically silent because meiotic success requires homologous cross pairing. The numerous methods to obtain haploid embryos of plants can be classified into 2 categories: in vitro-based method and in situ or in vivo methods. The in vitro-based methods are based on the culture of haploid cells and their differentiation into haploid embryos and ultimately haploid plants. Both male, in this case microspore or pollen, and female haploid cells, in this case megaspore or ovules, are used depending on the responsiveness of the cell in a given species. There are 2 main methods of haploid induction, including in vitro-based and in vivo-based haploid system. The in vitro-based system is based on the culture of immature male or female gametophytic cells. The in vitro-based methods are including in vitro-induced parthenogenesis or gynogenesis and androgenesis, in this case anterculture or isolated microspore culture. The in situ or in vivo methods make use of particular pollination techniques using irradiated pollen, interspecific crosses, or so-called inducer line. The second supporting technology is the genome doubling. For sterile haploid plants to become fertile, haploid cell lineage must undergo doubling. In this case, doubling the chromosome in the tissues or the DNA replication without mitosis. We can have artificial haploid genome doubling. Chromosome doubling may occur at either in vitro culture stage by using media containing the chemical to directly generate double haploid embryoids or callus, or at a later stage on the regenerated haploid plantlets. This technique is popular to genome doubling and mass haploid production by the use of colchicine, herbicide containing antimicrobial agent, and nitrogen dioxide. Then spontaneous haploid genome doubling. Spontaneous haploid genome doubling has been reported in many genotypes at low level for nearly as long as haploid have been studied. Single spontaneous haploid genome doubling typically reach at level approximately 5%. However, in some genotype have fertility level can reach 50%, for example, in rice. Spontaneous haploid genome doubling can be observed via semigamy, polyembryony, chromosome elimination, gynogenesis, and androgenesis. Alternatively, a pipeline can be developed that utilizes single spontaneous haploid genome doubling, thus replacing the use of a chemical doubling agent and permitting direct seeding of putative haploid into the field. The exact physiological time point of when spontaneous haploid genome doubling is occurring appears to vary and is unknown in most cases. After chromosome doubling that makes two identical copies of a single genome, each haploid chromosome doubled, and every gene is homologous or will be diploid plants, which are referred to as double haploid and a completely homozygous. Of regenerant from various haploid techniques. Haploid identification of related plants should be done in the first place to separate haploid from others. Example in rice, haploid sorted in no auricle, less vigor, with abundant small tillers, generate the spikelets containing degenerative anther and make haploid sterile and low in fertility. Then the double haploid similar to donor plant, normal spikelette, fertile, and normal seed setting. Cross between maize colors with haploid inducer containing R1 gene, which is purple color in embryo and aileron. Haploid seed can be identified as colors embryo with purple aileron. Artificial genome doubling can be done by using colchicine at 0.06% in 0.05% DMSO solution applied to cut coleoptile of haploid seeding for about 8 hours or overnight. Then the chromosome counting. Chromosome counting can be done during cell division either in mitosis or meiosis phases. Conducting chromosome counting at mitosis is easier and faster than that at meiosis phase. Only need root, tips, or other meristematic tissues. The third is flow cytometry. Flow cytometry measured flow level from early development when in callus or freshly regenerated plantlet. This technique provides faster, easier, and massive evaluation of ploidy level at early generation. Thus, make this technique is preferred than chromosome counting. For separating diploid from double haploid, we evaluate the plants through several techniques, including morphological evaluation, isozyme, DNA molecular marker, and enzymatic mismatch cleavage. How much does double haploid accelerate plant breeding? The development of large numbers of inbred or pure lines in very short period is a prerequisite for launching a competitive cultivar in the market. Double haploid technology supported the development of new cultivar through the obtainment of potentially large numbers of inbred lines with complete homozygosity in shorter times, which can either be released directly to farmers as new cultivars or used as inbred line for the production of hybrid seeds. In the figure, we can see that using double haploid technology, we only need about 2 growing seasons to acquire complete homozygosity, whereas by using the conventional breeding methods needs 5 to 8 generations to acquire almost complete homozygosity, about 99%. Furthermore, according to De La Fuente et al. 2013, in classical improvement in agronomic obtain successive selection so that we will achieve significant increase in genetic gain. The task can be accelerated if it is supported by suitable breeding method, reliable statistical analysis, and recently by using marker technology. While according to the variances, selection intensity, and parallel count of male and females and time. Breeding will maximize genetic gain by managing the components of this the equation, and the additive variance doubles while the dominant variance component of the phenotypic variance disappears in double haploid technology. Therefore, the genetic variance of double haploid is composed of only additive variance due to homozygosity at all loci, and the covariance between relative is twice that of non-double haploid individual. In addition to the changes in genetic variance, complete homozygosity of double haploid lines allows greater repeatability, which can reduce environmental variation through increased replication. Therefore, the response to selection during line development can be enhanced and the time required can be reduced by using a double haploid breeding scheme versus the traditional approach of making selection within segregating population. From genetic study of double haploids population, there is no dominant recessive relationship in using double haploid. The plants to possess a phenotypic stability due to the fact that all alleles are in homozygous state. Selection for dominant allele will be facilitated in double haploids. Recessive allele corresponding to them will not be found in the locus already occupied by dominant allele. Furthermore, there will be a reduction in number of population needed, an increase in probability of getting the side genotype because the number of individual plants in a population depend on the number of genes to be selected with assumption that they are unlinked. Using haploid method will need a minimum of 2 to the power of n plants. So if n is 3, minimum of 8 double haploid plants, while using classical breeding methods will need a minimum 4 to the power of n plants. So if n is 3, minimum of 64 plants. Selection efficiency is facilitated in order to obtain specific individual with genes of interest. Using haploid method, the frequency is half the power of n. So if n is 3, the possibility to get the side genotype may be 1 from 8 plants. While using classical breeding method, a quarter the power of n in double haploid technology increases the efficiency of selection, thus significantly reduce the cost, labor, and time in plant breeding process. Now we turn to the main method used to produce haploid embryos of plantlets. In general, anterculture or macrospore culture technique is even though the gynogenetic regeneration show high genetic stability and a lower rate of albino plants compared to androgenetic ones, gynogenesis, compared to genesis, a male gametophyte, is less frequently used for the production of haploid. It is because of the presence of, compared to the single gametophyte per ovule, and in part due to the ease with which anther and pure population of developing male gametophages can be isolated. For specific commodity, for example, in corn and potato, in planta haploid inducer line has been used lately. Used more than other techniques. Then we turn to factor influencing haploid embryogenation in in vitro based haploid production. The first is genotype of the donor plant that determines the efficiency of in vitro and in vivo haploid production. This response varies not only among being recalcitrant. For example, Brassicanapus is more responsive to microspore embryogenesis compared to Brassica junctia, and winter genotypes are more responsive than spring genotypes within Brassicanapus. In rice, japonica types are more responsive than microspore embryogenesis than indica. Is the physiological status of the donor plant. It directly affects the efficiency of haploid induction. Plant age also influences androgenesis. The frequency of androgenesis is usually higher in anthers harvested at the beginning of the flowering period and declines with plant age. Pollen from old sickly looking Brassicanapus and Brassicarapa yield more embryos than those from young healthy plant with anther from primary tillers in the growth condition of the donor plant significantly influence the physiological processes of their microembryogenic ability. Donor plant temperature and light condition influence haploid induction in wheat. Furthermore, regeneration response is usually high when the plants are grown under controlled environment condition, e.g. In phytotron versus field-grown plants. Third factor is gametophytic development. Microspore development in most crops is asynchronous, and microspore at different development stages may be observed in a developing anther. Time to collect the inflorescence, for example, in rice at putting stage of flower bud is critical. Bud size standardization is the critical step and is highly genotype specific. Hence, selection of inflorescence of flower buds that have maximum proportion of embryogenic microspore is essential for efficient microspore embryo yield. The state of microspore development at the time of culture initiation is the most important factor influencing the microspore totipotency during in vitro androgenesis. This occurs because microspore would only respond to embryo formation at a developmental stage when they are not committed to develop into pollen grains. Microspore can be switched toward the sporophytic pathway within only a narrow period. For most species, the period around the first haploid mitosis, in this case at late uninucleate or early binucleate stage of development, has been shown to be the critical stage for microspore susceptibility to androgenic induction. Fourth factor is pretreatment. Pretreatment, the explant before or after culture initiation, was beneficial in improving the efficiency of double haploid production in many crops. Pretreatment is needed to arrest the microspore in their gametophytic pathway. The development is triggered through embryogenesis by promoting cell division, followed by the formation of multicellular structures, and finally the embryo-like structure are released. The most widely used pre-treatment includes temperature, either cold or heat shock, and then sucrose nitrogen starvation and osmotic stress. In this case, mannitol or high sugar or sugar starvation, which differ among species. The fifth, the constituent of the basal medium and combination of the regulators. They play an important role in haploid induction. The reprogramming of X plant from gametophytic to sporophytic pathway, for example, depends on basal medium. Either a media like N6, B5, MS, etc., and the time and concentration of carbohydrate and plant food regulator will also have determining for the culture. Last one is culture incubation condition. It has profound effect on the induction and development of haploid embryos. External stimulus such as pre-treatment is needed to switch gametic cell development from gametophytic to the sporophytic pathway in many species. Besides pre-treatment, light intensity and light dark culture condition also play an important role in haploid regeneration. Now we turn to the application of double haploid and plant improvement. Haploids and double haploids have very important application in field of plant breeding, genetic, and molecular studies. Some of these applications are detailed as follows. Number one is rapid production of the pure homozygous line for variety improvement. In plant breeding, it is very much essential to get the pure homozygous line, which is generally obtained through pure homozygous line are used for breeding as well as genetic research. For example, in maize nowadays, haploid used to speed up hybrid development by public and private plant breeders to shorten the time it takes to generate a homozygous line from 8 to 2 seasons. This is arguably the latest major breakthrough in cycle time. Third is the significance in early release of varieties. As time taken for getting the homozygous line is less, so ultimately many new varieties can be obtained with less effort and less time by applying this technique. In Indonesia, using underculture of F1 from three-way cross, we obtained Bioprima agritan. It's crossed in 2015 and the line was released in the early 2021. Number 4, to obtain true to type mutant. It is important that mutant population are derived from an inbred or homozygous line, preferably double haploid line, to avoid the detection of false positive owing to inherent variation in the by using double is also easier. This method can overcome the masking effect of the presence of dominant gene. Then of induction of genetic variability. The pollen microspore are easy explant for production of genetically variable types by introducing the different foreign genes through different transformation procedure. The transform or transgenic haploids can be induced in the inbreeding program. Number 6, generation of exclusively male plants. In case of asparagus, a dioecious plant, male plants XY give more yield than female plants. Haploid production, the double haploid plant containing both Y chromosome can produce super male. Thus, haploid are produced from anther of male plants followed by chromosome doubling of the Y chromosome resulted in super male YY. The super male can be produce of XY. Number 6, accelerate gene pyramiding. For example, development of varieties with high yield resistant to biotic and abiotic stresses. Haploid production can be used for introduction of several genes of interest into cultivars. An established cultivar is crossed using double or three-way cross to obtain F1 and either F1 or F2 until the size.
Ah, moga-moga rezeki ya ya. Iki nih barang wis iso jeng. Enak.
Plant hopper, bacterial leaf blight, and blast were released in hybridization.
Pak Bambang, I think your microphone was mute. It's mute. I think Professor Bambang lost his connection. So please wait for a moment.
Hello.
Oh, welcome back, Pak Bambang. I can see you.
Is it okay for me to proceed?
Yes, yes, Pak. Please forward for your question.
Okay. Can it be seen? Can you hear me?
Yes, Pak. We can see your presentation.
Okay. Continue.
Yes, Pak, please.
So I arrive at application number 8. Yes. Okay. So double haploid can be very useful in genomic selection, but number of quantitative trait loci control a trait with low heritability. Simulation studies in mice showed that the responsiveness and efficacy of genomic selection based on double haploid line were far better than in marker-assisted recurrent selection or in F2 population with different number of QTL. The predictive accuracy of genomic selection was very similar to theoretical outcome within full sheep families, but also reflected the influence of sample size and trait heritability. Crossa et al. 2014 also reported parallel result for genomic predictive accuracy in their study of maize breeding at CIMMYT. Thus, double haploid lines are highly suitable for genomic selection with high predictive accuracy. I cannot move.
I think your connection has a very limited bandwidth. So
yeah.
Okay.
Sorry. Oh, start lagi.
Yes, it disappear again.
Well, So I for the example I just mentioned about the example of successful haploid breeding
So
in
can we
double haploid.
share your presentation from here, Pak Bambang?
Oh, yeah.
Can we
Okay.
help you to share your presentation from here?
Okay, please.
Okay, yes, Pak. Please wait. Yeah, can you see the material, the presentation?
Langsung aja, example aja.
What?
Langsung.
What pages? Terus. Next.
Number 23.
23.
22.
23. Was it? 23?
Yes.
Okay.
Yes. Okay. So, application of double haploid in marker-assisted selection program using either phenotypic or marker-assisted approach is conducted by replacing the self-pollination step at the end of the program to reduce the time for fixation of allele. The combination of mass and double haploid genetic gain and shorten the time required to cultivar breeding. If double haploid are used, fewer individuals are needed to identify an individual
Pak Bambang, we lost your connection.
Oh.
Oh, so I can hear your
Hello?
voice right now. So please continue.
Next slide.
Next
slide. Thank you. Okay. Example of successful haploid breeding and double haploid production and several important crops. In Europe, double haploid has been long incorporated into breeding program to develop commercial cultivar with horticulture crops and tobacco, and more than 290 varieties has been released. In rice angel culture, derived varieties in China has been released 20, Korea, South Korea 3, and Philippines 1, and India 8. In Indonesia, we obtain 3 high-yielding varieties from agriculture in Bari HDB Agritan, and then Bioprima and Biomas Agritan. Next slide will present double haploid production and several important crops. So just show quite rapidly for the slide. This is under culture of wheat. Next, please. Microspore culture of wheat. Next, please. Under culture in maize. Next, please. So this is the latest one for haploid inducer, has been routinely used in to induce double haploid production in maize. Next, please. This is rice underculture, which developed in cooperation ICGRB, IPB University, and in the Indonesian Institute of Rice Research. Next, please. This is underculture of eggplant. Next. This microspore culture in eggplant. And underculture in tomato. The detail probably you can read later. Underculture in pepper. Next, please. Then microspore culture in pepper. Next, please. Microspore culture in cabbage. And last one, underculture in tea. Next slide. So in conclusion, there are we have some that I will mention. There are two main categories of plant haploid induction, including in vivo and in vitro-based methods. In vivo-based methods of haploid induction consist of induction and regeneration phases. In plant breeding, and apart from maize, the production of double haploid plant requires at least an in vitro-based process, the success of which remains highly species and genotype dependent, as well as labor-intensive and time-consuming. By coupling rapid line development with homogeneity, double haploid breeding is ideal for countries which require high level of cultivar purity. Double haploidy is also ideal for rapidly responding to changing conditions, such as when a new pest biotype occurs. Moreover, the double haploid technology has not yet applied to all breeding programs, and there are still some major crop species, for example, soybean, tomato, sunflower, that are recalcitrant to the currently available procedure, or for which present protocols are not efficient enough. The main reason for non-inclusion of double haploid as primary technology for cultivar release universally could be its resource intensiveness, lack of skilled workers, and high initial setup costs. Hence, double haploidy will continue to be a tool for well-resourced laboratories that need to derive lines rapidly for selected crosses. It not be the main breeding method used, but it should be the method of choice for line and variety development. Thank you. And this work is in collaboration with ICGRB Bogor and IBB University. Thank you very much. I return to the moderator.
Thank you very much, Professor Bambang Purwoko. It was very thorough and concise presentation on plant genetic and breeding techniques. Ladies and gentlemen, without further delay, let's move forward for the second presentation. We would like to invite Dr. Sunil Arshak from National Bureau of Plant Genetic Resources, India. Please let me introduce you to Dr. Arshak background. Dr. Arshak completed his doctorate degree on plant science from University of Hyderabad in 2007. Dr. Arshak is very active in the international and national committee and working groups on plant genetic resources. You can further read through his CV in the program book. Today, Dr. Arshak will present his talk with the title Plant Genetic Resources Informatics for Enhanced Utilization of Plant Genetic Resources. Dr. Arshak, time is yours.
Thank you very much. I hope I'm audible and my voice is clear. Hello?
Yes.
Am I audible and my voice is clear and the slides are visible? Hello?
Yeah, it's clear. Yeah.
So shall I continue?
Yes.
Thank you. Thank you. Thank you. Good morning to one and all. First of all, at the outset, I would like to thank the organizers of second International Conference on Genetic Resources and Biotechnology. Thank you very much. In these hard times, you have continued to organize these conferences and continue to have the spirits high of the researchers and share the ideas and research outputs. Thank you very much for that. I also thank for inviting me to take part in this. Okay. As it has been just introduced, I am going to talk to you on a subject called Plant Genetic Resources Informatics, PGR Informatics, called in short. It's little more than just documentation of genetic resources. And the purpose I am going to talk to you on this topic is how can we enhance the utilization of plant genetic resources. Okay. It's more important to introduce the organization where I'm working than myself. It's because the work that has been carried out is in this organization. I work in National Bureau of Plant Genetic Resources, which is located in Delhi, which is a constituent organization of Indian Council of Agriculture Research, India. This bureau is the place where India's gene bank is housed. It is the second or the third largest gene bank in the world in terms of the volumes. Okay, and it's a very huge gene bank. We work on multiple crop species belonging to 13 crop groups, 1900 species. So you can imagine such a Herculean task, and it cannot be achieved by one place, one institution. So we have established 10 regional stations of NBPGR located in different agroclimatic regions of the country, because India is a huge country and different agroclimatic zones are there. So we are fortunate that any crop species that has been collected from within India or has been introduced from any other country in the world, we have a location to multiply, grow, and then make use of. So we are making use of this kind of endowment given by the nature. You can see that mandate of NBPGR, National Bureau of Plant Genetic Resources, is to act as a nodal institute at national level for all activities related to plant genetic resources management, from acquisition, that is from exploration from within the country, collecting, or from introduction from the other countries, to genomics-based profiling of indigenous and exotic resources. Our mandate is also, a part of the title of this talk is to promote the sustainable use of PGR. We also carry out capacity building programs through lots of training programs of young students, farmers, researchers from within India and from abroad. And we also have a master's degree and PhD program in plant genetic resources, PGR, which is a unique program. So we generate half a dozen PhDs every year who will then contribute in PGR management in the country. We carry out DNA printing of varieties, germplasm, and also we have a laboratory which is working on GM detection, genetically modified organisms detection for specific purpose. So you can see that NBPGR has a very huge mandate. It's very wide, as well as it is very deep. It's both width and depth. To tell you about the achievements and then link these achievements to the talk. In 2019, because I don't want to give you 2020 outcome, which has been truncated because of the COVID-19. In 2019, we carried out 34 explorations within the country, collecting 2,116 accessions, out of which 36% of the accessions were crop wild relatives. Currently, the focus is on crop wild relatives and trait-specific germplasm. In that year, 2019, 12,255 accessions have gone into the gene bank. 10,997 of them have been tested for seed health. The endeavor is to ensure that each and every seed in the gene bank is free of all the pests and diseases. At this point of time, in 2019, the seed gene bank contained 443,000 accessions, 444,000 we can say. And the in vitro gene bank has 1,800 accessions. Cryo gene bank has more than 500 accessions. Field gene bank has more than 5,500 accessions. And the medium-term storage, which is mainly used for the supply of the material, has 84,000 accessions. And we also have a National Genomic Resources Center where genomic resources are conserved, the clones and the genes and other things. So we have 397 of them. In 1 year of 2019, 145,000 samples that have been imported were quarantined. NBPGR also carries out the quarantine of the samples that are imported out as well as that go outside the country. Each and every sample meant for research in India has to come through NBPGR or has to go through NBPGR. That is the procedure in the country. And 29,000 samples meant for export were quarantined. 89,000 accessions were imported and 2,599 exported. 11,000 accessions were supplied to breeders and students in the country. NBPGR carried out 29 field days. These are huge field days when we carry out large-scale characterization and evaluation experiments of the crops. During appropriate stages, the farmers, breeders are all invited for these field days. They choose their accessions and then later they are supplied. We carried out 11 training programs and 236 publications came out in that year. Now, by this, you can imagine the extent of the activities as well as the scale of the activities. So it is very important that all these things are managed very well, systematically for current and future activities. So which is why information management becomes extremely important for us as well as all other gene banks. After this introduction, I would like to tell you that I'm going to present in 3 parts. First part is to introduce to you what is the subject of Pgr informatics and what extent we are working in India on Pgr informatics, and then what challenges we have in this area and what new initiatives are being taken up globally, and what is the future, what are the future activities. So let me introduce to you to the subject of plant genetic resources informatics. Is the top part clear? Because I see this Zoom's. How do I hide this? I don't know. Okay. We all are affected by climate change. No country is spared, and Southeast Asia is much more affected, seriously. So it depends upon the country, how do we respond to this climate change? If we are smart, if we are very well prepared, then we start early. Those who start early start with identifying the sources of genes in wild species that can make our crop varieties resilient to these climate change impact. So if we do that, then we work on wild species. And if we don't do that, we have less time. So we have to search for useful haplotypes in cultivated species. And if we don't do that, if we are still late, then we have to look for pre-adapted material that is available in the gene banks. And if we are still late, we are not good enough, then we have to look for existing varieties, whether in the country or outside, that are suitable for cultivation in these changed climate. And if we are sleeping still, then the country has to import food grain. So it is for the country to decide where we have to wake up and when we have to start taking action. Now, what is important from a gene bank's perspective, except this food grain import, in all other stages, the role of gene bank is very prominent. So it is not for somebody else, but the gene banks to proactively think about this, start taking action on these, and then advise all the stakeholders, whether it is breeders or farmers or government, to take action, to work on this. So because of this, now it is very imperative that these climatic changes, they ask for identification of prospective genotypes. Those prospective genotypes could be crop wild relatives or haplotypes within the cultivated species or germplasm lines or anything, to be fed to the variety development chain, which is present in different countries. They can be slightly different. These development chains can be slightly different. However, every variety development chain requires feeding of the plant genetic resources. So it is the responsibility of the gene banks to carry out this. Now, how gene banks can do that? You have seen the extent of activities without this, without doing any of these special activity. Already gene banks' hands are full. So, which is why it becomes very important to understand what different gene banks globally, countries are doing to respond to this. Okay. Now, everybody is, one thing is very sure. Each and every researcher of worth his salt, or each and every gene bank which is seriously thinking about this, is working towards exploiting the natural variation. There is no doubt about that. Only the procedures are different depending upon the facilities available, depending upon the training the person has, and depending upon the crop and the objectives. But everybody is looking to exploit natural variation. So there are different methodologies. Everybody is working towards this. You have seen the previous presentation also, how various tools are employed. So the objective is to carry out phenotyping, large scale phenotyping. For example, if Indian Gene Bank has got, say, let us 25,000 accessions of a specific crop, that's a wheat. Then you grow all of them in a single place, in a controlled environment, that means without any stress, carry out phenotyping, carry out genotyping of all of them, then study the population structure, carry out the genome-wide association, use various packages and all these things. Some of you are already aware of this, perhaps trained also in this. So once you carry out all these things, then we will come up with QTLs involved in various traits and sometimes genes. So the moment we do that, we are prepared. We are much prepared to become resilient. Our agriculture can be much more resilient. Not only this was, yes, everybody has been following this past 15 years or so, maybe 10 years in a much more intensive manner. Now the concepts have improved, new things are being added. Now what is called as envirotyping techniques is being used now. That means not just phenotyping or genotyping, it is very important that we also carry out envirotyping. So people are trying to find out environmic markers and they're working towards that. In simple terms, it is very important to identify two things. One, the origin of that particular genotype, because of which the adaptation has taken place, and the location where you are intending to grow a particular variety, where the selection or the stress is going to be there. So all these things have to be taken care. And this was in fact for gene bank purpose. This particular slide I'm showing here is from 2021 publication meant for breeding program. But this concept was there in gene bank management as well since long, in the concept of core sets. Create subsets of gene bank accessions based on the locations, the geographies, okay, from where they have been assembled. all the breeders in the world have been working on specific traits, but it is very difficult to work on a trait called adaptation because it's a complex thing. So that adaptation is provided, or a surrogate for an adaptation is from where it has been collected. So people started creating core sets. Slowly and slowly, the core set concept has, let's say, transferred from location to the trait. That means phenotyping based core sets you have from all the CG gene banks. And then from that, you have a genotyping based core sets, or you have a combined core set as well, phenotype plus genotype. We at NVPGR are trying to develop a concept, or trying to develop core sets based on a concept called dynamic core. For example, if a gene bank develops a core set from 10,000 of a 10% to, let us say, 1,000. But I as a breeder, or I as a PhD student, will not be able to work using 1,000 accessions. It's very difficult. Extremely difficult in case of cross-pollinated species. Very, very difficult. You cannot manage the area, the number of plants per accession, etc. It becomes very difficult, unmanageable. Then I should be able to extract from the information that gene bank already has. Let's say I can manage only 50. So I want to ask the 50 most representative accessions from the gene bank, which is called as a dynamic core. Or an institution has a larger mandate and a larger area facility, they would like to use more than 1,000. They would like to use 2,000. So they should also be able to use. So this kind of concept we are working towards, using both phenotyping and genotyping. This takes care of the envirotype concept. Okay, now, which is why it becomes very important, because all the students and young researchers are more attracted towards molecular diversity, using molecular markers, using genomic tools, etc. But that's not sufficient in gene bank management, or to enhance the utilization, or in fact, to begin the utilization. Only molecular data is not sufficient. We should have phenotypic data, that is field data, which is very essential. But what is also very essential is the local knowledge. Now, from whom you have collected, from where you have collected, what has been the specific traits in terms of their adaptation, specific uses, nutritional importance, all these things must be recorded. All these things must be recorded. Unless we link molecular diversity and local knowledge, including all other layers in between, we will not be able to enhance the utilization of these genetic resources. And that is the way forward for gene banks. So this is based on some work which we have carried out already. So you can see here that we use passport data, which provides us the location and the year of collection, which tells us what was the climate like in that year, how it was in 1960s in that area or 1980s in that area will be different. And information from herbaria, okay, for taxonomic identification and various other purposes. Information from weather data, information from, let's say, literature, information from phenotypic data, genotypic data, and all these layers have to be put on each other, use specific tools for this, which will then help you to map these specific accessions based on your specific objective. The moment you map them, like the one picture you have seen, you can see on the right side, these dots are specific accessions collected from these locations. And this area is the one which is in red is based on a specific statistical model, which shows that more accessions will be available in those areas. Now, this will help us in making specific decisions. Where to go for collection, how do we plan collecting explorations, and then where to do look for crop wild relatives. How do we take decisions for on-farm conservation? Do we have any specific areas which are rich in endemic species or haplotypes, which we have to take care, because that area is going to be affected seriously by the climate change? All these things. There can be multiple questions these specific information pieces can help make decisions properly. And this is being used regularly now. Now, I will ask few questions myself and try to answer very fundamental basic questions. Why PGR informatics? Why we should have a PGR informatics? To answer that, I have to tell you what is PGR informatics in a very simple terms. But before that, let's try to understand. A huge country like India, people are working in very good universities. Students are working in very good advanced universities, which are located in, let's say, cities. And there are some colleges which are located in rural areas. People are working, NGOs, lots of NGOs are working with farmers in some rural areas. Farmers themselves, some smart farmers are there who would like to work on their own, saving their seeds, selecting from their own germplasm, or looking for new and better one. So there are various stakeholders. Now, it is very important that a gene bank should take care that every bit of information that it can give it to stakeholder must be available to each and every stakeholder. And an online system, an organized digital information system, provides fair and just opportunity for each and everyone to access. And this is the fundamental crux of any information management system. An information the basic purpose of an informatics system is to ensure that a person in an advanced college or a person in a remote area will have equal opportunity depending upon the facility that they have, but they have equal opportunity to access. From your side, you have not graded the access. Just imagine if all of those things are there in your library, published in books and catalogs, they are there in limited areas. No, they will not have access. So it is very important that everyone should have access. And please note, best gene banks have smart databases. If you want to work systematically in all of your activities, have very smart, advanced, ready to let the use by plug-in kind of databases for each and everybody to use. Okay. Now, the PGR informatics can be very simply defined as management of diverse information related to PGR. Management and analysis of diverse information. Now, management includes creation, storage, retrieval, and presentation. These appear very simple English words, but each one of them has a separate set of data managers. It's not a very simple thing. Every step requires a skilled person, a set of people, and analysis, discovery, exploration, and extraction, depending upon your use, the user. Whether you are looking for extraction or exploration or discovery, one should be able to do that. And now, because we have advanced computers, computing ability, and internet facilities have advanced over last few decades, extremely, internet especially. Now, facts, figures, statistics, knowledge, news, photographs, videos, you just name anything. They all can be combined and put in data, which was not possible. When I started working on databases, it was simply not possible. When people started working on MS Office, Microsoft Office, initially, it was impossible to copy from PowerPoint to paste in Word or from this to that. It was impossible. Those young people, you will not realize this. Now it is very easy because you have got XML files. You can copy from anything, from anywhere and paste anywhere, which was not possible earlier. Now that this is possible, we should make use of this and create most comprehensive databases. That's the purpose. Now, what information? This is a very redundant question because I just said every information. Name anything about an accession. So every information related to that. No information is insignificant for a database. No information is insignificant. Today, some information might appear insignificant. After 10 years, it will become extremely important. So for a database manager, every information is significant. So passport, import, seed health, I have mentioned some of these. And some of the arrows at the top do not have names because the information types keep adding. Genotypic information, genomic information, sequence information, okay, and then SNPs, whatever you just name, they all keep getting added. Sometimes we have farmers information from where we have collected this specific one. Some of these text or videos are in local languages. In India, we have many languages. So these are available in local languages. It is very important that you record information in the local language, because the information, the syntax of the names provide you the trait of the genotype. And if you translate that into English, it will be lost. So like this. So all information is very important. And we carry out that. One should ensure that it is up to date, accurate. I'm not going to explain all of them. Reliable, systematically stored, and easily accessible. The last one is extremely important. If the user finds it difficult to access the exact specific information that he or she wants, then the purpose of this entire information system is defeated. Okay. Now this is very important. For whom we are going to manage this information? For whom? Now, basically, three classes of stakeholders are there. The first and foremost for a gene bank person is the gene bank managers themselves. Now, just imagine if a curator has to take out accessions for regeneration. He or she has to quickly know, without any doing lots of efforts, that which are the accessions that have been conserved more than 15 years ago that have to be taken out for regeneration. So they should get on their email or their specific system, these are the 105 accessions that should go to the field. So accordingly, they will manage their logistics, book for their field, carry out the regeneration. Okay. How many have to go for evaluation for, let's say, viral resistance in a specific crop? Okay. So that has to be done based on the information that is already there. Please remember, the information management is a cyclical one. That means somebody will collect information from the database, generate information while they're working, and this information goes back to the database, which becomes an input for somebody else. Okay, so this is very important. Now, second kind of stakeholders is breeders and plant researchers. Their objective is to choose specific germplasm plant for their work. Now, the objective or the purpose of Pgr informatics is to enhance the choice. The greater the choice, the greater is the ease of choosing, the greater is the utilization. So that is the fundamental purpose at this point of time in NVPGR. How do we enhance? How do we give a wider variety of choice for the breeders and plant researchers? We also have a process called plant germplasm registration. This is nothing to do with the new varieties. New varieties also you can register as a new plant variety in some other organization in India. But I'm telling about plant germplasm, such as genetic stocks. You have identified some specific genotype which are high oil content. It is not good for growing as a variety, but it has got this specific gene or QTL or a specific haplotype which is providing it very high oil content. So it has to be conserved, and it has to be, the person who has identified this has to be recognized. So this is a soft IP, intellectual property. The moment that is there, then it gets popularized and more people start using that one in their breeding programs. The information system also has to cater to third set of stakeholders, and that is policy makers and funding agencies, because allocation of funds, political approval of programs, all these things depend upon what work you have not done. It's not important that what you have done, what you have displayed, how much information is available to others. So the moment we start building an information system for NBPGR, we have to see that we have to have different modules for all of them to cater to their purposes. There are some international agreements, conventions, etc., which require each country, whether it is you decide for your own purpose or not, if you are a signatory to any one of these, Convention of Biological Diversity, and part of that is Global Plan of Action and International Treaty. If you are part of all these processes, it is a must on the part of the country that you generate a specific information system of your own and then make it available to people. How much you can disclose and how much you need not is up to you. Okay, we don't have to discuss at this point of time. Okay, today we have excellent information systems available globally. Very, very excellent information systems. For example, Genesis is a portal. It's not a database where primary data are stored. No, Genesis is a portal where the information from all the CG gene banks, CGIAR gene banks like IRI, like CIMIT, okay, all these gene banks are available at one place. So Genesis is that from ICRISAT, which is in India. Okay. Apart from that, many national gene banks have also shared their data with Genesis. So all these things are available at one place in one format. So Genesis is becoming more and more popular over months now. We also have a Eurisco, which is also very fantastically developed database. It's one information system. Unlike Genesis, where basically passport data and some amount of trade data is available, Eurisco is a very complete information system. Eurisco also has got lots of genotypic data. Japanese Gene Bank has in Naro has got its own information system, which is also very well maintained. You have got to trade data on the crops that are conserved in Japanese Gene Bank. We have got a GRIN, which is of USDA, American Gene Banks. Although now USDA has shared its data with Genesis, they also maintain their own information system called GRIN. Okay, these are ones which are very fantastically maintained, updated very regularly, and they're very useful. Those who are interested in material will certainly go to them, choose, and then indent, ask for the material. One can do that. Now, compared to that, I have to tell you where are we standing in India, Pgr informatics in India, and what work we are doing or what work we have done over the years. In India, NBPGR is the nodal organization. So whatever Pgr informatics work in India that has been carried out is the responsibility of National Bureau of Plant Genetic Resources, and I am taking care of that as a head of that particular activity. As I mentioned in initial slides of introducing NBPGR, we carry out a number of activities like any other gene bank: conservation, evaluation, characterization, acquisition. Acquisition includes two sets of activities: import, introduction, or exploration and collecting. Simultaneously, we also carry out quarantine process. We have a national herbarium of crop plants, which is a recognized herbarium in the global herbarial organization. We carry out DNA profiling. There is a division on genomic resources, independent division consisting of about 20 scientists. It's a huge division working on genomic resources. We have a set of people working on biosystematics. We work on climate change. We work on policy and IPRs. So you can imagine that all these activities have to be included. If you want to develop an information system for NBPGR, all of these activities have to be included, linked, and updated regularly. Means something happens in one, it influences the other. So you have to develop a very comprehensive and dynamic information system. So this was the requirement when we set out to develop information system for NVPGR. You can see how the condition was. You are conserving 300, 400,000 accessions in your gene bank, and everything is in paper format. You have files, etc. It's impossible to search for something very quickly, or sometimes impossible. So you have to convert from here, which was there in 1950s, to convert it to a catalogs kind of thing, then transfer it to digitization, and then you must have an information system. So it's very important, and we are happy that we are here and we are not in this stage. Okay. When we started, we had three options. We clearly had three options. Number one, at that time, USDA, along with Biodiversity International, and later along with Crop Trust, have developed Green Global, which is a system which CG gene banks are using now. Green Global. Green Global is a system. Either we could jump into Green Global, but we had some policy issues. Government of India, decision makers did not agree with sharing all the information that we have. So that was an option which was not agreeable. And later it turned out that they were not ready at that time. Okay, in 2010, they were not ready, around 10 years ago. They were not ready with the Green Global to be shared with everybody. Second one was very obvious. Yes?
I think you have 5 minutes more for your presentation.
Okay, I'll do that.
Okay.
Okay,
sorry,
sure.
Thank you. Yes.
No problem, no problem. Either we could outsource or we have to develop our own. So we decided to develop our own. So in 2010, we started doing all these things. And you can see that slowly and steadily we started developing a number of web apps, applications, mobile applications for all activities that I mentioned just now. Each and every activity we have a mobile application. Usually, when I talk on this particular subject, my basic objective would be to demonstrate all the applications to the audience, to the people who are attending. But at this point of time, unfortunately, because of the COVID situation, we are in lockdown. Our institution is working with few people, and our servers are all shut down. So I'll not be able to demonstrate, but I'll just tell you where it is there. So Pgr databases, one can visit on our website, or this QR code is there. You will be able to reach there. We have this national Pgr portal, what is called as Pgr portal. Just Google Pgr portal, you will come to this Pgr portal. And here you will get all the information on the passport data of all the accessions that are available in the gene bank. And it is open access. You don't need any login. No application from NVPGR requires any login. All these are open access. Anybody from anywhere in the world can access. We have many other apps. You can see Gene Bank, Seed Gene Bank, Cryo, In Vitro, MTS, Field Gene Bank. We have specific applications for all of them, each one of them. Similarly, to enhance utilization, we have different kinds of applications for other purposes. Okay, we have a map-based selection. It is a touch-based one. You just open in your mobile phone or a tab. It works very nicely. So if we have developed all these so many applications, what has happened? You can see here our Pgr portal applications access from various parts of the world. So you can see here this data is from Google Analytics. So people from lots of places in the world are accessing the portal that we have developed. So it is pretty useful for everyone to find out information about the material that is conserved and available in the National Gene Bank. In a few years ago, 2016, we organized the first International Agrobiodiversity Congress. And because of these applications and impact, we had a separate session on Pgr informatics. So it was a very successful one. This year, we are second IAC is being organized. Some of you must be knowing already. I am part of the Genesis Advisory Committee because of the impact that we have generated based on our information management system. I am also a member of the Global Information System Committee, Scientific Committee. Okay, all these because of the work that we have done. So quickly, I will tell you about challenges and future. Data quality remains challenge for all the gene banks, and local languages. It is very important that we learn quickly how to convert everything into local languages. Anybody who wants to use in his or her local language, they should be able to select, and then everything gets connected, including syntax, and which is a great challenge, not very easy at all. We are working towards that. New initiatives globally are, first and foremost is digital object identifier, in which Indonesia is working in the forefront. You have already organized many meetings, and we're working towards that. You have trained people also. People globally are trying to work from descriptor to annotation, how to convert these to this, so that we bring both phenotype and genotype together. How to work with wild species and crop wild relatives is a challenge. People are working towards that. And how to work at an ecosystem level, and how to work not just on Pgr. If you're working on a specific geographical area, how to document all the genetic resources in one place is again a challenge. Okay. We are working towards crowdsourcing applications to start working on-farm conservation, custodian farmers, and community participation. Okay. And there are other works, including gene bank index. Okay, just take a couple of minutes. As all of you know, plant genetic resources work is always a teamwork. One person cannot do it. It's not a laboratory work. So a whole network is required. So I acknowledge all my colleagues, present and past, who have generated so much of information data, which we are able to convert it into a specific format and then generate lots of applications. And it requires funding because we need people. So fortunately, I have received funding continuously from one or the other source. And I acknowledge each one of the funding source. Right now, I am a national fellow, which is a national professorial chair, specifically on Pgr informatics. No other place in the world has a specific funding like this for Pgr informatics. So I'm thankful to Government of India through ICGRB that this specific activity is being funded. And with that, I thank you for your patience.
Okay. Thank you very much, Dr. Sunil. Due to the limitation of the time, we have 15 minutes for discussion. So we will read 2 questions for each speaker, and please answer the question concisely. So the first question and the second question for Professor Bambang from Dr. Dani Satyawan. One of the problem of haploid breeding is insufficient number of recombination, so linkage drag can be more of a problem. Is there any trick to deal with this? Also,
I have lost the connection. Is it me only, or others are also not able to hear this?
Yes, there appears to be a problem.
I'm sorry, I lost the connection. So I will read Then 2 questions for Professor Bambang Purwoko and 2 questions for Dr. Arshak. The first question for Pak Bambang from Dr. Dani Setiawan. One of the problem of haploid breeding is insufficient number of recombination. So linkage drag can be more of a problem. Is there any trick to deal with this? Also, has there been any attempt to develop rice lines that can act as haploid inducer similar to barley? The second question for Professor Bambang from Dr. Sri Kurniati. By using of double haploid method and producing plant, can this increase the expression of the gene target if the first step through transformation, then followed by double haploid? And then for Dr. Arshak, let me see from this step. Dari ini ya. Oke. Dari ini dari. Dari ya. It is from Miss Andari. May we know how your gene bank handling thousands of accession in the field gene bank and how much the average cost of the management per year? The second question, for the concept of dynamic core, is it mean that the core are instructed depend on the user preference? Is there any standard procedure or mechanism to develop the dynamic core, especially when the gene bank has also induced the environment adaptation or local knowledge of the Pgr? Thank you. Please respond to the question. Thank you.
Shall I start answering or? Professor Bamban is answering.
Okay, Dr. Sunil, please respond.
Yeah. To the second, I'll ask answer to the second question regarding the dynamic core. If based on the question, I understand that the person who has asked question is aware of the procedure that we follow in to develop a core. There are more than one procedure, but all of them specifically use the same statistical principle of using the best possible smallest representative set. Now, in dynamic core, what we have is, for example, you have you go to that our site, open, for example, in Rice, and there are, let's say, we have around 110,000 accessions of Rice. The moment we complete characterization of all these, the data will be available, and that we ourselves generate a core information, and that core set is also information available. Now, that will be a huge core set. We also have a genotypic data, or SNP data is being generated for Rice, okay, the entire set. So we are going to have all that in a couple of years. The objective is that the person has to go and say that my requirement is only 200 accessions of Rice. So you select 200 and run that same statistical procedure that we have run to generate a core of a standard size, but you will run it for a smaller size. So obviously you will miss some of the alleles. There is no doubt about that. But you will get a best representative of that one based on both phenotype as well as genotype. So that's the procedure. It will run on our application. It will give you information. I hope I have answered this question satisfactorily. And the first question was not very clear to me. If it is there in the chat box, I will read it and then understand.
The first question is, may we know how your gene bank handling 1,000 accession in the field gene bank, and how much the average cost for the management per year?
Is this related to cost of the running gene bank?
Yes, I think.
Because your voice was cut in between, so I couldn't get it. Sorry.
Yes, I think the related to the cost of management per year.
The cost, okay.
And how to handling 1,000 of session.
Yeah, I can't give you the figure right now offhand. I'm sorry, but I'll respond through email to the organizers on exact cost. We have the cost of that one. Okay. The major cost is the electricity cost.
Okay. Yes.
Yeah, for major cost is electricity cost.
Yes, I know.
Sorry.
Okay, thank you. Okay, thank you very much. So please, Pak Bambang, to respond to question for you.
Okay, thank you. First question about insufficient number of recombinant in barley. Well, at least in rice that we work on, sometimes we found also genotype dependent for the regeneration. So the effort that could be done, I think, increase the number of cultures so that we can increase the regenerant and obtain more double haploid that can be obtained. If it is it cannot be done, then it is not quite feasible to do it for double haploid methods in this case. The second questions from Dr. Koniati about the transgenic, whether it is increase the expression or not by doing the double haploid production. Double haploid actually rapidly fixing the gene of interest. So I think in the homozygous state that it can be detected or expressed quite rapidly. However, in terms of expression, it should be, it depends on the construct itself. For example, the use of enhancer, etc. So I think the key point is that we use double haploid method to rapidly fixing the gene of interest in the form of homozygous lines. I think that's my answer. Thank you.
Thank you, Professor Bambang. So we still have 3 more minutes. So I will deliver 2 questions to Dr. Ashok first. The question from Marilyn Ferrer from the field Rice. With this large number of accession you handled, how many researchers and laborers you have? How much is your average annual budget? It's similar from the first question, I think. And who are your funding source? For the second one is how NBPGR provide or gather all those important information. NBPGR works together with various research institutes in India, and how in your national plan in doing that, or the National Center of PGR doing that all. Thank you. And for Pak Bambang, I will read from the NEO from DC International. Is possible for haploid genome doubling to occur after interspecific crossing without chromosome doubling agent? If so, is it common or high chance? And what could be factors, or based on your expertise, interspecific crossing must be followed by chromosome doubling step in order to obtain the explant? Thank you. And the second one is from to Pak Bambang. Is there any certain criteria for scientists or breeder to decide the use DH in the breeding program, and how is the genetic stability of the new plant varieties generated using this DH technology? Dr. Sunil, please respond.
Memang sama. Jadi untuk interspesies itu juga sama.
Hello, Dr. Sunil, can you hear me?
Hello.
Yes, I can hear you.
Yeah,
Please respond.
Professor Bambag is answering, or should I start?
You can start first.
Yeah, thank you. If I have understood the question properly, because I'm reading from the chat box as well. One question is about how are we working with our a network of institutions.
Yes.
We have a huge number of commodity-based institutes in India, which are under the ICAR, Indian Council of Agriculture Research, ICAR. We are also part of ICAR. So we have network projects, very well-funded network projects, where a specific institute, specific scientists, or a team of scientists are identified, and objectives are set, funds are given, and we have project staff employed. So it's like any other externally funded project, we run that one. So we run it for 5 years, and objective is, let us say we have got 18,000, 19,000 accessions of germplasm of a specific crop. Then entire set of that germplasm is completed within that 5 years. Specific institutes will work on, virologists will work on virus resistance, entomologists will work on that, biochemists will work on biochemical profiling. So like this, individual activities are taken care by different people, and work is completed. Right now, for example, we have a $250,000 $250 million total worth projects from central government to carry out genotyping and some sequencing, resequencing of huge number of crops. So thanks to the funds that we receive, we are able to do this in a network manner. Now it's very important that if you don't have funds, if they have to work from their own funds, it's impossible. It's in a project mode. So that's the answer for that. And the other question was, there was clarity, lack of clarity for me, sorry.
Yes. So please, Professor Bambang, to respond.
Okay, thank you. I don't catch the complete idea about the questions. I hope that this will answer. Yes, for interspecific cross, then we have to double either by it depends on the species. It can be propagated first, and then we treat it with colchicine or herbicide that I mentioned before. Some of them, I think, including amipropos methyl, oryzalin, and pronomide and trifluorine. The second one, I could not catch. Would you repeat, Ibu Moderator?
Yes.
The second question, what is it? I could not. Maybe the connection is not that good, and
so the second is still from this setting. Is it, if so, is it common high chance? And how could be the factor? Or based on your experience or expertise, crossing must be followed by chromosome doubling step in order to obtain DH plant.
Yes, that's the first question, right? Yeah, I think.
Okay. So the second question is from Puji Lestari. Is there any certain criteria for scientists or breeder to decide the use DH in the breeding program? And how is it the genetic stability in the new plant variety generating using DH technology?
Okay. First of all, of course, depends on the crop. when we decide on the crop, then we will find the which one is established on the method. Of course, for rice, I think it's quite established method. And a second question, whether it is stable or not for double haploid. Yes, it is. We have experience before, probably you know Dr. Priyatna Sasmita working on rice with double haploids. We characterize from generation, I think generation 2, 3, and 4, and we find no difference at all from several genotypes that we characterize. So I think once you get double haploid, then it's homozygous, fully homozygous, that it will be, what do you call it? It will be transferred to the next generation, the same genetic constitution. So I think it's quite stable from generation to generation. That's from our experience for rice, at least in this case. I hope answer the questions.
Thank you, Pak Bambang. Ladies and gentlemen, I think we run out of the time, so it's already 10:51. So we actually committee allocated me for until 10:30. So we will deliver the remaining questions to the speakers and we'll forward the answer to you through the email. So we conclude this plenary session. Thank you for all participants for your time, and we hope you find this session valuable and stay healthy. Let's give the round of applause for the speaker.
Yeah, thank you. May I have just 5 seconds, please?
Okay,
yeah,
please.
Please send all the questions. Would you like to please send all the questions so that we can send answers?
Okay,
yeah.
okay. Thank you.
And thank you very much.
Yeah,
thank you very much. You can see that I'm wearing Indonesian shirt from my last visit. From my last visit to Bogor.
Okay. Thank you.
Very good.
And I see Sabran, and I just want to say hello to Sabran.
Yeah.
I miss all of you in physical meetings.
Yeah, okay. Thank you.
Thank you very much.
Yes. See you next time. Yeah, have a great time. So I give the time to Madam MC. Goodbye, everyone.
Thank you very much.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
Thank you, Mrs. Ranti, and the valuable invited speakers. I hope everyone had a productive discussion today. Now, before we start the parallel session, PT CropLife will share information to us. I would like to pass the floor to Mr. Siang Hitan, the expert. Okay, Mr. Tan, welcome.
Thank you very much for the kind invitation, Madam Chair. I'm trying to share my PowerPoint. If you can see it, do let me know. Is it visible?
Yes, Pak, we can see the screen.
Okay, thank you very much. I know I have 15 minutes, so I'm going to, again, nice to see Pak Bambang again online. Unfortunately, unable to go to where we can usually meet. So we're going to talk about plant genetic resources in the plant biotechnology space. Again, we've been working with CropLife Indonesia. We're working with the Bogor University, etc. So CropLife Asia is a regional association that works very closely with the farmers. Our members are BASF, Corteva, Syngenta, Sumitomo, Bayer, FMC. So again, thank you very much for allowing us to share with you today our work in the region. So for CropLife Indonesia, similarly, we work in various areas. Also, we work in technical regulatory. We do a lot of workshop together with government officials, anti-counterfeiting issue. We have issues in BRABES, etc., working together with DOA and MOA on apprehending some of the counterfeiters, stewardship training with various stakeholders in Indonesia, and biotechnology and seeds, again, technology, bring technology in the hands of farmers in the best timeline together under science-based evaluation. So biotechnology is one of the main strategy that we work in, especially on the seeds and biotech also. So but Alex, who is here today, is one of our coordinator and is spearheading this. So coming back to the original discussion, from the onset of life, whether it's from Darwin, from Mendel, the biodiversity is going to be core for us to be, just now our researchers talk about the plant genetic resources. This is the core, and how do we conserve them, and how do we move critical gene set into the breeding population. So again, from pure synthase, from anywhere, even Indonesia, from the oil palm. I come from an oil palm background, and I used to work on breeding in oil palm, more on the molecular side, so understands the oil extraction rate, and also been doing corn breeding in Pakistan and also Cambodia, and also work on a bit on hybrid rice prior to this. So again, the genetic resources, be able to get the latest yielding plant set into the hands of farmers is going to be critical, because we all need to feed more people with higher productivity, with lower resources, lower input. If you go even to Central Java today, whether the water resources is going to be a constraining issue, and whether we plant 2 corn and 1 rice and rotationally, it's going to be crucial. So we really need to have good genetics material into the hands of farmers. And across the region today, despite COVID, the growing population, the water supply and land, as I mentioned to you, I've traveled to, I used to run a plantation in Medan, corn plantation, and water supply is a core issue. Climate change and pest and disease is also core. I'm going to talk a bit more about pest and disease later on because of my lab, my previous work on genetics. So all of this is going to be crucial for us to continue to make safe and secure food and affordable food to our population, and continue to support Indonesia's agriculture GDP as both domestic market and also for exporting market. So United Nations is talking about feeding the 9 billion people. We know this, all of us are very familiar with the challenges from sustainability concept, from climate change adaptation, from pests. I'm not going to dwell a bit more on it. But on the crop and biotechnology and plant breeding, and again, learning from Pak Bambang today, and we have our CG people talking about hybrid vigor. And today, if you look at hybrid vigor itself, even in corn, in Thailand, 90% of their corn cultivation is all hybrid. Fundamental, if we don't have the hybrid vigor, we wouldn't be able to achieve. Even Indonesia today, the BC that you see is looking at 5 over metric ton, 4 over metric ton. Without that hybrid and the continuous breeding population that is available in Indonesia, the whole gene set that is being utilized for whether it's what you call hybrid, to maintain that hybrid vigor, that is very crucial, and to shorten the breed cycle with technology. So again, biotechnology is not new. We just celebrated post 25 years of adaptation of biotechnology. You look at US, 95% of adaptation, Brazil 94, Argentina 100%, Canada 90% also in terms of corn, canola, soy. So on the right-hand side of the chart, you see all the percentage of the areas that are being utilized for corn, canola, and soy. And fundamentally, soy is the largest genetic modified cultivated crop today. So this is rather a technology, and it has contributed significantly on the adaptation and mitigation, whether it's soil conservation with zero-till, water moisture conservation, and totally they have bring benefit to almost $224 billion to 16 to 17 million farmers, and 95% all come from developing or smallholder farmers across the world. So even like if you look at Philippines is a good example, with almost 800,000 hectares, all are on corn since the onset. They're all biotech-related corn, Vietnam, there's also biotech-related corn. So this is something that the whole of Asia is a corn negative region. We really need that corn for our animal feed industry, whether it's for chicken feed, it's for poultry, and etc. It's critical that we have self-sufficiency in terms of corn. and Indonesia has been talking about various other biotech components. You have your drought tolerant sugarcane also. Those are very important crop that is looking at, because you look at Thailand itself, because of drought and etc. If they have that biotech related corn, their productivity for corn can easily be moving from a 42 metric ton per hectare to at least 60, 70 metric ton per hectare if they will have the technology in their hands. So again, there's opportunity for development technology. There's so many researchers on board here. I just want to take a bit of time to talk about, it's 2 slides, is tobacco mosaic virus. We know this virus is detrimental, and they first discovered way back in early 19th century. And until today, it was first discovered in 1886. Only in 1996, the gene was discovered for the disease resistance. So time is crucial for pest and disease management. And this was done in, I'm happy to say that this was done in my lab in Japan, whereby it took us, a PhD student, almost 7 years to discover the locus. This is not even isolating the gene set and putting into the tomato plants that we want to. It took 100 years for us to get to that location. Whether it's marker selected breeding, etc., we all use it very often. Just want to give you the context why technology is important for us to quantum leap and get to the disease platform. And now with plant breeding innovation, we know where the location of the gene, we can go in and really isolate, excise, and integrate, or whether it's point-based mutation that we look at. The other piece that we looked at is also, this is tomato link. This is a second gene that we isolated from tomato, also done in my lab. So the other piece that we look at is also looking at terminal flower mutation, is to cut out flowering so that we could use in crops that are producing paper. Because the moment you have multiple apex growing, you'll reduce the energy level. And for paper pulp industry, you want a plant that doesn't flower and therefore keeping the whole plant correctly straight. So again, this is opportunity. It doesn't mean that corporate companies will have to do this, but this can come from a laboratory or a lab in university. So that's why the private partnership comes into play. How can we bring technology from university into fruition? So again, the gene editing model is there that we can work with, and globally, we are putting in about $6.5 billion in R&D every year. About 3.5 billion goes in the biotech and seeds arena. So these are some of the examples that today it's really commercialized as like waxy corn with high amylopectin production with SD1 mutation, non-browning of white button mushroom. You heard about what you call corn with reduced phytate content, and then many other crops are coming out from either a laboratory-based or a company-based. And that's why I wanted to share with you. So a lot of people have this misconception that it is dominated by a single sector, but unfortunately not. So a lot of countries have put R&D into developing new gene-edited products. This is just to show with you how much scientific publication has gone to rice, tomato, maize, wheat. And apart from that, in different crops also, from soybean, looking at agronomic value, food and feed quality, biotech stress, and one of the early CRISPR-Cas in corn was able to withstand abiotic stress like drought and still be able to maintain its 5 to 7% yield increment and protecting the crop. And this is where our farmers needs today in terms of adaptation and mitigation to climate change, because everywhere you're seeing either a stress crop because of water is not coming in, or even water that cannot go out, meaning flooding. We're seeing so much flooding everywhere. So without proper irrigation, our farmers needs more environmental adaptable crop so that they can fully harvest their product if any of the climate change do attack them or then they face. So just want to share with you, again, the landscape of US crop enabled by genome editing, and also to show you the democratization of technology in wide varieties and their benefit. So From the crop base, there's soybean, there's tomato, corn, pennycress, camellia, canola, rice, even down to avocado. And whereas in terms of trade, it goes into live shelf storage. Can you just yesterday, we just had a discussion on the total loss of post-harvest in Pakistan because farmers aren't able to protect their crop post-harvest. It runs into billions of rupees every year. And apparently the loss is about 50% in Pakistan for even some of the grains that they have. So for developing country, even in Cambodia, the loss of post-harvest storage is exceeding 35% for, from processing all the way to post-harvest. So there are a lot of opportunities like pest resistance is about 30% if you don't have proper management on the ground, yield improvement, stress tolerance. So going back to the last piece, when we talk about the democratization of technology, it is now not only my member company, look at it, there are small startups in US from Simplot, Yield Bioscience, Cebus, Inari, Corteva, of course, is our member, Toolgene, Benson, Crosscast, Evogene, etc. So this is where the opportunity for our R&D, whether it's from Bogor University, for anybody who has a technology, you're able to bring it to market with the speed to target the characteristic that is happening or impacting our Indonesian farmers. So we should open ourselves into adopting with fundamental regulatory and available. I think the researchers that are sitting here today in the conversation today will benefit from the democratization of the technology. So public sector, you have Illinois State, Florida, and etc. Just also to capture, today, there are 1,600 startup companies in the plant science sector. In Israel alone, there are 400 companies that is involved in agriculture startup. So it is a very golden opportunity for us to be able to have, with a functional regulation, I'm sure Indonesia's agriculture domain will flourish and will really expand because you have so many fundamental good breeders in around, whether it's Pak Bambang, you talk about haploid and etc. There are a lot of genome program on ground, and whether it's genome sequencing to marker selection, assisted breeding, there are so many gene sets that are available that we can take on and put into the varieties of plant that Indonesia wants to work on. So therefore creates the genetic variation that we need to build a larger germplasm for us to develop new plant varieties and provide addressing the genetic, what you call disease resistance or even drought resistance through different gene sets that are available. And not forgetting that all palm, I work on all palm as I mentioned to you, and there's a lot of genome and proteome genomics that we are working to look at selection at the embryo level after tissue culture to look at reduction of abnormalities in those sense before it goes into cultivation. So there are a lot of genetic tools available that both Indonesia R&D situation and also the university to benefit and bring technology and new varieties into the hands of farmers in a faster pace. So plant genetic resources is core. There are a lot of genetic program, and also don't forget the germplasm that is widely available in Indonesia could be adapted and integrated into the new what we call varieties that you may be developing through better technology. So, but Alex, my 15 minutes is up. I hope I managed to get it in time for you and open for questions. Thank you.
Okay, thank you, Mr. Tan. Ladies and gentlemen, now we enter the quiz. Let's have fun for a while and grab the goodie bag with Mr. Kurniawan. Go, 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 Mr. Kurniawan.
Okay, thank you, Dr. Roning. Good morning, all participants. I'm Hakim Kurniawan. I'm coming to you again in this second day of ICGRB conference to guide a quiz game, second ICGRB quiz. Well, in the first quiz yesterday, only few participants joined in this quiz. We hope that in this quiz today, we will have more participants that join in this quiz. Let's take a look in the screen, how to join the quiz. To participate in this quiz, you can use both your mobile gadget or your laptop that should be connected to the internet. The first one, open your Google search on your mobile gadget or your laptop, and then type the word kahoot.it. And then execute the kahoot.it link showing in the Google search results. After successfully get into the Kahoot, enter the PIN. The secret PIN will be shown in the screen later. Then after that, enter your unique nickname. Now, then you are ready to take the quiz. When the question is previewed in the screen, you can select the symbol that corresponds to your correct answer. Okay, again, in this two-day quiz, there will be 6 multiple choice questions. You only have about 20 seconds to answer each question. In the end of the quiz, we will select the first 3 participants who earn the highest score. We promise that there will be a very interesting prize gift that will be available for these 3 winners. Okay, are you ready now? Let's start the quiz. Now we have 8, 9 participants that already joined, 11, 12. Come on. Hurry. 14 participants, 15. $17. More participants. Okay, 23. Okay, come on. We're still waiting for the other participant to join in this quiz. And strictly, I have to inform that the participant that already taken as winner for the first quiz, it's not allowed to participate in this quiz again. We are sorry. Okay, now we have 30 participants. Where are the other participants? We have actually 89 participants that join in the Zoom meeting. 36. $37. We will start the quiz in a few minutes. $37. $38. $41. Hurry up, please join the quiz. Where are the other participants? Okay, shall we start now? Here we go. This is the first question. ICGRB is stand for, please choose your correct answer. We have 14, 16, 18 answer, 20 more. Okay, time is up. The correct answer is the International Conference on Genetic Resources and Biotechnology. There are 26 participants that taking the correct answer. And now who has the highest score? Anne. Congratulations. Next questions. The second ICGRB 2020 event is opened by, please choose your correct answer. Okay, time is up. The correct answer is Director General of IARD. There are only 17 participants that taking the correct answer. And now, who is the participant that taking the highest score? Soy. Next questions. Invited speakers who presented in the first day plenary session is, except, please choose your correct answer. Okay, the correct answer should be Bambang S. Purwoko. There are 30 participants that taking the correct answers. And now, Soy is still taking the first place, the highest score. Next questions. Invited speakers who presented in the second day plenary session is, except, please choose your correct answer. And the second day plenary sessions, except. Okay, time is almost up. Time is up. The correct answer should be Hiroshi Ezura. There are 26 participants that taking the correct answers. And now the participant that taking the highest score is still Soy. Congratulations. Yeah. Okay, next questions. The second ICGRB 2020 is discussing the current developments related to, except, please choose your correct answer. Please hurry up. Time is up. The correct answer should be big data management on genetic resources and biotechnology. And there are only 12 participants that taking the correct answers. And who is the participant that taking the highest score now? Oh, still Soy. Great. And now the last questions. These following are the public services managed by ICGRB at this moment, except, which one? Choose your correct answer. Okay, time is almost up. Hurry, hurry. Okay, time is up. And the correct answer should be assessment of agricultural technologies. And there are 21 participants that taking the correct answers. And now the final position, we have 3 winners. The third one is Win. The second one is Rini. And who is the first positions? Oh, still Soy. Okay, congratulations for these 3 winners. Please contact the organizer as soon as possible to claim your prize. Okay, thank you for your participation. And I'm turning now to the next session to the second ICGRB organizers. Thank you.
Okay, thank you, Mr. Kurniawan, and congratulations for the winners. Ladies and gentlemen, now let's start the parallel session. We will divide you into two rooms. There are Dendrobium and Edelweiss. If you want to go to another room, send a private chat to the organizer, and we will move you to that room. Okay, today's parallel session will be led by Dr. Surya Diantina in the Dendrobium class and Ida Rosdianti, MSc, in the Edelweiss class. Now, I would like to hand over the floor to the two of moderators. Thank you. And now, finally, we enter the closing ceremony of this conference. Now, the chair of Indonesian Biotechnology Consortium will present his closing speech. Dr. Siswa Setiahadi, welcome.
Thank you very much for inviting us, KPI. So, Honorable Director General of Indonesian Agency for Agricultural Research and Development, Bapak Dr. Fajri Jufri, and respected the Director of Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development, Bapak Mastur, our distinguished invited speakers, distinguished Director of Partnering Companies, and our dearest participants. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, ladies and gentlemen. We finally come to end of this conference. We are pleased to be able to contribute in this excellent conference together with the Indonesian Center for Agricultural Biotechnology and Genetic Resources Research and Development, ICGRB. We hope that the collaboration does not stop here, but will continue in other scientifically important events. We also hope that this conference will encourage collaboration between participants and their respective institutions to advance research and to solve crucial global public problems in the field of genetic resources and biotechnology. In this virtual meeting, we recognize that there are problems such as with the internet connection and other unfortunate technical obstacles. We sincerely apologize for all these unintended and unanticipated problems that occurred during the conference. I would like to express my appreciation to our respected invited speakers, presenters, and participants for their contributions during the conference. Additionally, many thanks to the sponsor, PT Crop Life, PT ITS Science Indonesia, PT Fajar Mas Murni, and PT Prima Instrument Analytica for their support in this conference. I also extend my gratitude to all committee members for all their hard work for this conference. I hope that we can harness technology for conservation and sustainable use of genetic resources for food and agriculture from this conference. Now I declare that the second international conference on genetic resources and biotechnology is closed. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Setiahadi, our collaborator. Okay, ladies and gentlemen, this is the end of the conference. We are very grateful for being together with all participants in this conference with tightly scheduled and had virtually unexpected things. After this conference, don't forget, we still stick each other because the process of full papers review will be started, Scopus proceeding publication. Also, a lot of thank you for full supported sponsors. Hope we could again in the Third ICGRB. Stay healthy and goodbye. Wassalamualaikum warahmatullahi wabarakatuh.